apologize for delay in starting. We have some technical glitches, so finally we resolved this, so we can start the program now. So I am Professor Siddiqui, and I'm sharing this session with Professor Dr. Atiyul Wahab. It is an honor and really it's a pleasure for me to introduce her. She is one of our star scientists at the International Center of Chemical and Biological Sciences in Karachi. She is an amazing person. You know, she actually got her PhD in structural biology. Then she got some training in organic synthesis, and then she switched to molecular biology. So that's amazing to have. Professor Dr. Atiyul Wahab, currently she is a professor at Yes, University of Karachi. She joined the institute in 2010. She completed her PhD in organic chemistry in 2007 at HEJ Research Institute of Chemistry. Afterwards, she undertook a postdoc in structural biology under the supervision of Professor Dr. He was a Nobel laureate at the Scripps Research Institute of USA. She did a postdoc at Dortmund University, Germany. More than 80 research yes, patents. One work on solving problems in mass spectroscopy. Two edited monographs on of field survey in the entire world. He has also supervised 37 472 Pakistani scholars. Is protected scientist from PCSD in 2015 and 2020. At national level, Professor Dr. Atiyul is leading NMR user. She is leading NMR user group and excels in the scientific instrument program. So please welcome Dr. Atiyul.
Assalamu alaikum. Good morning, ladies and gentlemen. Thank you so much, Dr. Rafat, for very kind introduction. So I will be discussing about an application of NMR spectroscopy, which is saturation transfer difference NMR spectroscopy. Actually, my lecture was supposed to be after the lecture of Professor Iqbal Chaudhry, who was going, but due to his engagements, so he's going to deliver the basic concepts of NMR spectroscopy, but due to his engagement, my lecture comes first. So application is first, and then you will be listening the concepts, the basic concepts of NMR spectroscopy. So I'd like to introduce my supervisors, from whom I learned a lot. Professor Taur Rahman, I got PhD under his kind supervision. I was working in his lab, and he was minister at the time, but he was so passionate. So whenever he come back to Karachi, he always have to discuss with all of his students. And we are always in pressure. We have to give a very brief synopsis reports of our research to him. And Professor Iqbal Chaudhry, he has constantly guided us in not only the research work, but also I learned a lot from him, all that spends in my life. And for structural biology, I got training under supervision of Professor Good.
Excuse me, ma'am. Can you hear me?
Yes.
Ma'am, your slides are not shared for online.
Okay. Where is Zoom? No. Okay. Okay.
Read this thing and more. It's floating.
Yeah. Shimshad, can you see the slides now?
Yes, ma'am. Thank you.
Thank you. So my training after I got my PhD in molecular and structural biology was done under supervision of Professor Kurt Rich, and from whom I learned all the basic concepts of structural biology using NMR spectroscopy. I also had a couple of postdocs under Professor Harald Schwalbe in Goethe University, Frankfurt. So this STT NMR spectroscopy actually ligand-based detection of the part of the ligand that is interacting with the protein or enzyme. So it's very important and a very sensitive technique, as we all understand that proton NMR is the most sensitive nuclei in NMR spectroscopy, and this is proton NMR based detection. So it doesn't require any kind of labeling of samples, which always cost a lot. So during this technique, we saturate enzyme or protein with a train of Gaussian pulses, and at certain point of time that we need to select, and then there is a cross relaxation phenomena which through which the this pulse is being transferred all over the molecule, and then those molecules which the ligand which is bind with the protein also got the transient anode from the molecule, and that is transferred to that part of the ligand which is interacting through transient anode, and then we subtract this on resonance and off resonance spectra, and we got the difference NMR spectra. So that's what saturation transfer difference NMR spectroscopy. So here is some spectra, the carbonic images that explain that when a ligand is binding to the protein molecule and we saturate protein through any point, we need to select by different experiments, and then these molecules bind with the protein, and those parts here you can see these A and B are the part of the ligand that are interacting with the protein molecule, actually got the signal and in the difference NMR spectra. Not changing. Yeah, not working. Yeah. Yeah, it's fine. I'll do it. Thank you. Yeah, so these are the steps that we use for the saturation transfer difference NMR spectroscopy. We first record the proton NMR of a protein and just a simple proton NMR to find out the resonance where we would like to saturate and give the pulse for saturation. And then after after we find out where we would like to select those points that which is at least 1 ppm away from the ligand molecule or ligand signal, so that there should be no false saturation transfer difference we could observe. And then when we select different resonance point and then run the STD spectrum again only with the protein to find out the maximum saturation points which the protein has. So sometime where we can see the maximum saturation. So then after running different points only with protein, and then we select the highest saturation point, and that point is selected for the STD spectrum. We also run, these are the control experiments. So we also run the NMR STD NMR experiment for buffer, just to avoid and check that any component of the buffer is not showing any false STD effect. And we again do the STD NMR experiment of ligand only. Again, these are control experiment. Just wanted to show that there is no false STD we are going to observe. And then we run the real STD experiment with saturation transfer with compound and protein. These are the standard pulse sequences, STD diff, which is present in Bruchel. 0.3 version we use, there's 0.2 and 0.1, and the saturation time usually, it depends to number of protein, with usually 1 to 3 seconds. But the relaxation we give the maximum, because we want to make sure that whatever the pulse we are giving, everything need to be very much relaxed. In Bruker pulse sequence, is by default they can have like 5-second relaxation time, but maximum usually in the experiment we give 1 or 2 seconds, but here we give 10-second relaxation. So we want to show that whatever the pulse we have given, before giving the next pulse, the system is already completely relaxed back. So, and it depends, number of scans also depends the concentration. So usually with the cryoprobe on 600, we have 512 number of scans. It could be 1K. It depends on the number of the concentration of the ligand we have, because it's ligand-based techniques. We are not going to observe any signal of protein. So we also, I'll be discussing about cell-based. This was, I explained you, which is pure protein and ligand interaction. We have also done cell-based STD NMR experiments. And again, there are all these control experiments that we have to take, because we have used this technique to for one of our project on aromatase inhibition, and we have prepared the microsomes from human placenta, from the maternal side of the placenta, which has been discarded. We collected from the hospital and prepared the microsomes, and these microsomes have the highest concentration of aromatase enzyme, which plays an important role. The overexpression of these enzyme plays an important role in breast cancer. So we use these microsomes and find using STD NMR spectroscopy, and find the cell-based interaction with the small molecule. And we also do group epitope mapping, and by using this technique we find out which proton is in the closest proximity of the protein, because what we do in this one, that we have the saturation transfer difference NMR, difference we got from on resonance subtracted from the off resonance spectra. Off resonance, usually we saturate because there are two parallel spectrum. One is on resonance, in which we give saturation to protein, but at the same time we want to subtract the original spectrum. So off resonance spectrum, usually 25 or 30 ppm of the proton NMR spectrum, where there is no signal we observe. So so the difference NMR, looking at the integral values, we see which proton has highest integral value, and we give it 100%. And then the other proton are relative to that 100%, sometime like 45 to 64%. These are some examples. So relative to the 100%, we give the other proton based on their proximity. So that's why this is group epitope mapping we do using STD-NMR spectrum. And we also, these are the different techniques we employ. So since my lecture is only on STD-NMR, so I try to explain what we are doing in our institute. And this is the determination of KD by using saturation transfer difference NMR spectrum. And here we titrate ligand with the protein. Once we have some molecule which is interacting with protein, and we usually in STD-NMR, we take like 100 to 200 times more concentration of ligand as compared to protein. So if we have 10 micromolar protein, we will take 1 millimolar of the ligand molecule. So ligand concentration is usually very high. That's why we observe only the ligand protons. And for the determination of KD, we titrate with different concentration. Usually, for example, it depends, whatever, for example, if we are using 1 millimolar concentration of the ligand, then we will start down less than 1 millimolar and more than 1 millimolar concentration. So here there's an example. Here we have used like 0.25 and 0.5, and then 1 millimolar, 1.5 to 2.53 millimolar concentration. So these, because we are facing lots of problems, these are small organic molecules which are mostly water insoluble. So what we do, we usually make a stock of the ligand with 100 millimolar concentration, and we take, if we have to use 1 millimolar sample, so it means 550 microliter is usually the 5 mm tube with the 5 mm NMR probe. We use 550 microliter of NMR sample. So we add 5.5 microliter of ligand dissolved in DMSO. So we would like to consistently using a particular concentration of DMSO. We don't want to have very high concentration of an organic solvent that could disrupt the activity of protein. And once we do this titration, and this need to be calculated for each proton. So for example, if we are calculating, we would like to calculate one proton. So we determine the STD amplification factor using this formula. And then the STD amplification factor of all protons need to be calculated in the titration. And then we take an average for the protein to determine the KD. And this amplification factor, then we can calculate the KD using this formula. And And for this one, this is one of the technique that using NMR, we can determine the dissociation constant of a ligand with a protein. And to supplement our experiment, we also do fluorescence-based thermal shift assay, in which we can determine the binding of ligand with the protein. And it's the cypro-orange dye that we use usually. So protein concentration is very sensitive, you know, fluorescence is very, very sensitive experiment as compared to NMR. So only 2 micromolar concentration of protein is enough, and with almost 20 micromolar concentration of the ligand. And we use cypro-orange dye, and this experiment is run on a real-time PCR. And the increment usually is given from 20 to 95 degrees Celsius with an increment of 0.3 centigrade per minute. So with here there is the TM, the melting point of the temperature, is being calculated. So what we do here, we record the TM of the protein only, and then we also add ligand in the protein and record the TM. So if the TM is changed, if the molecule is destabilizing the protein, so the TM of the protein should decrease. Or if it is stabilizing, so the TM of the protein will increase. So based on this one, we determine the binding of a protein and ligand and its stability. So by plotting this one against temperature. And this is also one of the experiment to determine the KD using the fluorescence based thermal shift assay. One experiment I just explained you by using NMR spectroscopy, and this one is being done using this is same thermal shift assay. And here again, the temperature is being plotted with the log concentration of the ligand. And so it's again the titration experiment to determine the KD. So we have to use various concentration from 0.1 micromolar to 1000 micromolar concentration, but it depends the concentration of your ligand in STD NMR that you have used. So a little bit less and a little bit more concentration have to be used. And so different concentration has been used, and this is being plotted. This is the concentration of the ligand in nanomolar against the TM. So here the log KD can be the intercept is the log KD, and when we take the anti-log of this, so we get the KD in micromolar. Okay, so now these are the basic techniques that we use in our institute for ligand-based screening. And one of the example, we have used the repurposing approach of US as FDA-approved drugs against SARS-CoV-2 main protease. So we all understand that drug repurposing is an approach to find out the new therapeutic indications of already approved drugs, and it reduces the time and the cost for the drug discovery and development process. So SARS, there are two proteases important in SARS-CoV-2. One is AMPRO and the other is PDL-PRO. And here I would like to explain that these two proteases are playing important role in the proteolysis after the formation of the polypeptide. So this, if these are inhibited or at this time, so at this stage, so there will be no non-structural protein, and subsequent pathway will be stopped. So these are valid targets for the treatments of SARS-CoV-2 main protease. So Dr. Iqbal asked us that instead of using, NMR is a very expensive technique. The NMR time is expensive because the maintenance of NMR requires continuous filling of liquid nitrogen and liquid helium. So it really, NMR time is very expensive. To save the time, what we have done, and this was as per the instruction of Professor Iqbal Chaudhary, he guided us that instead of running single molecule STD, we have to make a mixture. And then we tried lots of different things, and finally we were successful. That we usually now make mixture of 5 compounds together and run through STD NMR spectroscopy against different enzymes. So here in this one, we have evaluated 155 drugs and compounds in 31 mixtures. So that reduced the time a lot, because we are running STD NMR experiment of 5 compounds together. We already have the proton NMR of each one. So if we find the STD effect, we compare the difference NMR with the existing proton NMR of each compound. And if we find, okay, this, then there we can discover that this particular molecule has an effect with STD, but there might be some limitation because these molecules, when they are together, they might interfere also with the binding of the protein. But we have to compromise. That's why we can to use this technique more efficiently. And we have evaluated 31 mixture, and out of which these are the compounds, 10 drugs that shown the interaction with AMPRO. And with these identification using STD-NMR, my colleague, Dr. Sabah Farooq, she is virologist, and she is propagating SARS-CoV-2 in Vero cell line. So this assay against the antiviral assay was done by her, and using this assay we could see that these compounds are showing some effect on the virus propagation. So these are the propagation of the control cells, Vero cell line, and these are the cytopathic effect after 24 hours, 48 hours, and 72 hours. And these confirmation has been done using the quantitative real-time PCR, and we identify ORF and gene and E gene, and to find out that virus is really propagating in these cell lines. So chloroquine phosphate, this is also recommended for the treatment of SARS-CoV-2. So we have used this as to find out as a standard, and here we can see that I'm sorry. So we can see here that there are this part of the ligand which is interacting, and as H19, H20, H14, H16, and these are the plaque reduction assay, which shows that at 82.3 micromolar concentration, 100% reduction. And so that's why it's been diluted half to 41.5 micromolar, and it gives 63.2% reduction. And we have further done some docking studies as well for this one to see the interaction of ligand with the protein residues. Okay, so again, at remdesivir, it's also been reported, and here we could see that these are the color one which are showing some interaction. Different percentage of interaction, the color coding explained, and it also gave 86.7% plaque reduction with 82.3 micromolar concentration. And this is dexamethasone. Here we could see these are the protons which are interacting. I'm sorry, yeah. These are the protons which are interacting with the protein. And here we could also observe this inhibition. And this is one of the drug which is used as a calcium channel blocker, lutetium hydrochloride, for the treatment of high blood pressure and hypertension. And here we can see that these are the metals here which are interacting with the protein in different NMR. And the plaque reduction assay also shows 81.8% inhibition. And these are the docking studies which shows the interaction of small molecule with the protein binding site. And these are the dynamic simulation that we have conducted. And the blue one is the protein protein RMSD and the red one is the ligand RMSD. So here you can see that after 20-25 nanoseconds, these are not very much stable, but is the stability observed after this is 100 second, 50-second is simulation. So after this, the complex was more stable. And this is mefenamic acid. Again, we could observe these are the two protons, two methyl group, S16 and 15, which showing interaction with protein. And mefenamic acid is a anti-inflammatory drug used to treat painkillers. And this is the plaque reduction assay, which shows 81.8% reduction in the viral plaques. And these are the docking studies, which shows the binding of ligand with different residue, leucine, glycine, histidine of protein. And these are the ligand and as ligand and protein RMSDs on 50 second simulation, a nanosecond simulation. And here we could see they are quite stable complex. And lorcetin, potassium, which is angiotensin receptor blocker and used to treat high blood pressure. It also shows these are the protons in different colors, which shows the interaction. Here you can see this one, this CH2 get the highest, we give like 100% because this shows the highest integral value, and all other are relative to that one. So these are not real 100%, but relative to that. And this the plaque reduction assay, which shows 54.5%, and these are the docking scores. Again, the simulation studies. And mexi-lentin hydrochloride. This is also one of the drugs we have checked on STD NMR, and we can see these are the protons that are interacting, and the docking studies and the simulation. And amantadine hydrochloride is also shows almost all these protons are interacting. And here 72% reduction, and the docking studies and simulation, and this is the volume calculation we have done, and which shows that the blue one is little bit closer, while the gray one is a little bit farther. And this is the TM that I have explained you, that the calculation of TM using fluorescence assay, and we could find that glucine, HBr, and trimine butane malate are the one which are destabilizing the protein a lot, because this one is AMPRO itself, and these are the ligand with the protein interaction. And this is the oral plaque reduction assay, the comparison, and you can see that lost in potassium has shown quite a lot less reduction, and the highest reduction are observed with 86 and 88% with remdesivir and dexamethasone. And then again, we used one of the same experimentation for dengue virus protein, which is NS5, a non-structural protein of 5. And so these proteins are not available in the market. So we cloned it in our lab, express it and purify it, and then use STD NMR. So lots of molecular biology before this spectroscopic work. So dengue infection is very common in our part of the world. And according to WHO report, over 390 people million got infected annually. And 20,000 people die with different stages of dengue. And according to WHO, 2021, 48,000, more than 48,000 cases been reported, 183 deaths in Pakistan. But there are much more cases, because these are the ones which are those people which go to the tertiary care hospital. But there are many which don't go to these hospitals. So these non-structural protein has the two domain. One is RNA dependent RNA polymerase domain, and the other is AMPTase domain. So the polymerase works for the polymerization of RNA, and AMPTase do the methylation. So we selected, this is very important target for in Flaviviridae family, and we have selected this target for the expression, cloning, expression, and purification of this protein. And then NMR studies, we have evaluated 75 FDA approved drugs in 15 mixtures, and 10 were identified that could be lead to for next step as a potential antiviral agent. So this one is diclofenic sodium, and this diclofenic sodium here you can see these are the red one showing the highest percentage, and the other which is H12, and the other is being calculated accordingly. So different percentage of STD we can observe, and these are the docking studies with the NS5 domain. And this is another drug which is etoposide. So we have done the same methodology. We have made a mixture, first analyze the mixture, and then identify the compounds that are interacting with the protein. And these interaction, then we take the individual compound and run a separate full STD spectrum, because this the short mixture STD and NMR experiment, we usually take like 128 number of scan, which requires about 1.5 hours, and because this is difference NMR, so on resonance and off resonance pulses. So, but for the full STD experiment, we usually 512 or 1K number of scan we take to get a good signal to noise ratio. For the mixer, we just wanted to see if they are really interacting, any molecule interacting or not. So that, and that's why we do, we try to save lots of NMR time. And this is atenolol, and here you can see this, this one, the 9 is assigned as the highest integral value, and then the rest is being calculated for this. And these are the interaction of docking interaction with the protein. You know, I'm a chemist, but I always do real-time work. So I for docking, for me, docking is like to give beautiful images. I don't trust it all. But it just gives a nice representation, because you know, if whatever you give to computer, they give dock it even if there is no real interaction. So first we see there is an interaction, but to present nicely we just do docking as well. But we already know there is an interaction. We don't give like docking as the only parameter. So that's what I learned from our teachers, Professor Taur Rahman and Professor Iqbal, that we want to see something really happening. And so this is another drug that we have find out, and that shows, these are the red one, which shows the highest interaction with the protein, and other is optimized accordingly. And another molecule, which is another drug which is interacting, here the STD NMR experiment and the docking score. So after this, we would like to see that, you know, these are the interaction of molecule with protein. It doesn't show that it is inhibiting, because this is not an inhibition assay. So we have used to find out if this molecule is really inhibiting RNA-dependent RNA polymerase, and we have done the polymerase inhibitory activity only those selected compounds that we have selected after STD NMR experiments. And here we have used the template, and these are the primer, and it's we incubated at different time of interval, and what we see, we find that out of all these, diclofenac sodium is the one who is really inhibiting the activity of RNA-dependent RNA polymerase activity at 500 and 1000 micromolar concentration. So here in this gel, you can see these are the 9 nucleotide, the regular product. There is sometime the misincorporation, we can get like, you know, these are smaller bands with the 10 nucleotide due to misincorporation. And these are incubation time with 0.5 hours to 1.5 hours, a different concentration of diclofenac sodium. So here we could see the activity is being decreased. So this is not only done with the full protein NS5, but we also checked it with the individual, only the RNA dependent RNA polymerase domain. Just wanted to make sure that it you know, without AMPTase domain, if the activity remains the same or it changes. So for in-cell NMR, STD NMR spectroscopy, there is one project which we have been doing for the identification of aromatase inhibitors under supervision of Professor Taur Rahman and Professor Iqbal. And we have identified many aromatase inhibitor which plays an important role for the breast cancer progression in Pakistan. The breast, it's second world in the world. Pakistan is the second number on the breast cancer, so it's really an important area for us to do research. and what it plays. So what is actually happening? So aromatase is the enzyme which convert the testosterone to estradiol, and or androstenedione to estrone, and these are the one which increase the proliferation of aromatase of breast cancer cells. So if we inhibit these enzymes here, so estradiol and estrone production will be decreased, and subsequently the proliferation of the cancer cell will also decrease. So these are the three clinical drugs which are present. One is exemestane, which is substrate mimic, and these are the two non-steroidal, estradiol and letrozole. Actually, we have, through biotransformation, we have identified many potent aromatase inhibitors, which are much more than anastrozole and letrozole and exemestane. But here I'm going to present something which is non-substrate like. So through biotransformation, we got the substrate mimic. So we acquired the human placenta of the maternal side, which is being discarded after delivery, and these are we got the ethical clearance, by the way. So without ethical clearance, we can't work on an in vivo and human samples. So these aromatase is being chopped in small pieces, and then after several steps of ultracentrifugation, we got the microsomes, and these microsomes has the higher concentration of aromatase enzyme. And then we have optimized a fluorescent assay in which we can, this is 96 well plate based assay, in which we can calculate the inhibition of these drugs against aromatase. So these are the letrozole standard drug, which shows 14 nanomolar. It's very active, and these are the STD. So here, this is in cell. So we are using microsomes for STD NMR. And here, this is the proton NMR of letrozole, here 1 millimolar concentration. And these are the proton NMR of letrozole plus microsomes. And this is the STD difference spectra, here you can see, and of difference spectra of letrozole and microsomes. And with the buffer we usually don't observe, but we record all these control experiment in case if we see something, we have to subtract it from to get the final difference spectrum. And this is anastrozole, which is 9.4 nanomolar activity, IC50 value. And here you can see these all are proton which are interacting in STD. And the saturation, again, the highest integral value has been given 100%, and then rest is being divided. And we have again used the human placenta for docking studies, human aromatase for docking studies, just to show the interaction. And here we have find out that there is this molecule is usually fit very well and within the active site. And this is again, these are the sesamol, the small molecule which shows interaction. Here you can see these are the interaction we could observe in STD difference spectrum. And this is benzocaine, which is again a small molecule, and we could see the interaction of all protons here. And these are again a small, very small molecule, toluidine hydrochloride, which shows different level of interaction. Then one more thing that I would like to show you, that we use NMR to see the reaction product. So this is one of the enzyme, which is NADPH dehydratase enzyme from vancomycin resistant Staphylococcus aureus. And this is metabolite proofreading enzyme. Again, we clone and express and purify these enzymes. And this is ATP dependent, ATP ADP dependent. And this enzyme dehydrate S form of NADHX or NADPHX by utilizing ATP or ADP. So here you can see NADH is being converted to R and S of NADHX. So here, this is the point where you can see these other R and S product are formed, but this enzyme NADH dehydratase dehydrate only the S to NADH, and these are further cyclized to form cyclic NADHX. So this enzyme perform specifically dehydration on the S form, and here these R and S form are interconvertible. So in cell, whatever the product is formed, the S will be converted. So it also play an important role in oxidative stress. And here you can see this is like a functional assay we have performed, and here you can see the proton NMR spectrum, which is recorded of only this molecule, and the 1D-toxy experiment. 1D-toxy is again a very beautiful experiment. It's the experiment in which you can see the whole spin system. Since you can see this molecule is this is so many protons. So if you want to see the whole spin system, you can use 1D-toxy. You can just see different spin systems on different pages, which makes things very clear. We used to do it during I used to do it in my PhD. It's when we wanted to identify different sugar components. So it because sugars are very much overlap. So 1D-toxy is very beautifully separate all these. And here you can see this. These are the S, R, and the cyclic. This is cyclic, and this is the S form, and this is the R. Here we can observe. So we have done this experiment in the NMR tube, and we added all the reaction mixture in the NMR tube, and with the time dependent. And again, we have identified that only this particular proton, which is S form, is being dehydrated by the enzyme, while the R form and the cyclic will remain as it is. So these are the superimposed images. We have already been published all these work. So in conclusion, NMR is a very robust technique for structure determination, ligand protein interaction. But this is again only the ligand observable technique. We are in our group, we are doing the chemical shift mapping, which require the labeling of the protein. It's again the titration experiment, but using 15 NHS QC, but it requires labeling, and labeling is very expensive, and specifically if protein need to be labeled like doubly labeled or triply labeled, so it costs really a lot, because we have to use glucose, labeled glucose, 13C glucose, as the only source of carbon for bacteria to grow, because we used competent cells, different type of competent cells of E. coli, and 15 ammonium chloride as the only nitrogen source for the bacteria in the media. And sometime for triple resonance experiments, we have to use instead of normal water, the media has to prepare N2O, which really cost a lot. So labeling is really expensive, and so this technique is one of the beautiful technique in which we can find out the ligand and protein or enzyme interaction. And tent. So I'm really fortunate that I have two great mentors, Professor Taur Rahman and Professor Iqbal Chaudhary, from whom I have learned a lot, and I'm keep learning. I still remember when I have given my PhD viva. So Sarata asked me, how are you feeling? I said, sir, I'm much relaxed. He said, you shouldn't be relaxed. This is just the starting point. So you have to move further. So whatever your teacher says is just always remain in our heart. So, these are the 13 superconducting NMR spectrometers we have. Couple of years ago, all the system is being upgraded to the new system NEO of Bruker, which is the highest technology. So the sensitivity is really, really very high of all our spectrometers because of the new electronics. And we have 4 cryoprobes, the one 800, 2600, one on 500 megahertz. And we have multi-nuclear probe in which we can detect all type of nuclei, ideally all nuclei, but it takes time, depends on the concentration. And thank you so much for your attention.
Thank you very much, Dr. Wahab, for an excellent presentation. I don't know very much about NMR, but I've learned a lot from your presentation, how it can be very useful for guidance direction. So thank you very much. I think we have time for questions.
Can I ask?
No, you go ahead.
Very interesting things that you want to say. We live in all this it's been used by the interactions in protein, you can really provide more appreciation. Although it's a prediction, but there's a lot of work we published and this establishment. And then now I think to me it's quite new to be using NMR. So it's not prediction, as I mentioned, it's not prediction, it's confirmation, cumulative indication of interactions between the protein, the ligands, and the compound with the protein. But I believe both methods can be used. In fact, the computer is usually preliminary, yeah, and then followed by using an NMR we should just like the experimental in vitro your work proof by looking at the response.
Yeah, actually, you're right. I'm sorry. I don't want to offend anyone, but I just said what my true feeling is. So, as a matter of fact, one of our colleague, she's been doing this. She has, do the docking studies first, and then out of these docking studies, she tried to, based on the docking score, then do some other in vitro examination. But there are, it's like, for me, it's like, if you take something which is really low docking score, and if you can select something in between, it's like the random screening. So you may find some hits also. So definitely you can use it, but it's, I just been trained to see something real.
So based
Yeah.
on following on his question, because the reason why computer aided design was accepted because random screening,
Yeah.
the library too many
Yeah.
difficult.
Yeah.
So from the computer aided design studies, you filter out some compounds, and then to see if indeed they are binding, you can do your experiments, and then you go to the assay.
Yeah.
Finally.
I
They should just complement each other.
Yeah.
Yeah.
Yeah, but in my opinion, if you those you just eliminate from the docking. If you select randomly from those, and you will find definitely from, and we have done this because I asked my colleague this from these this in which she has been using Schrodinger software for this computational studies, and then I asked her that she has to select those which has the higher docking score, and you haven't selected for the screening, that randomly choose those. And in random, she also finds some molecules that that shows interaction in STD-NMR. So it's yeah, it's I don't want to offend anyone, but I really appreciate, and it's just for those who are working on computational studies, and it's really nowadays the computational study is really move ahead, and lots of good things are happening, and there are some drug also in the market using that has been shortlisted from computation.
Thank you very much. I have some practical questions.
Yeah.
When you do the STD-NMR, of course you are changing the concentration of the drug when you do the titration, and when you do the STD, you focus on the proton NMR of the drug, right? But of course you have already done some NMR about the protein or for the protein. Are there some requirements for the concentration of the protein you are using?
Yeah, usually the protein concentration is very, very low.
Yeah.
The bigger the protein gives the good STD effect because of the transfer of cross relaxation phenomena. If you have like more than 30 kDa, bigger than 30 kDa protein, you get very good STD effect, and the concentration is
how much?
Yeah, concentration usually if the protein is more than like around 100 kDa, like 1 micromolar is good enough. And if around 30 or 35 or 40 kDa, maybe 5 or 10 micromolar.
And your
so not
deuterated solvents or 50%
deuterated solvent.
So
we made buffer in deuterated solvent. Yeah.
Thank you.
So only deuterated solvent we use for this effect.
Thank you very much, Zia. It's amazing lecture and the integration, the work with different techniques. I believe every technique has its own. It's okay. And every technique has its own limitation. So
Yeah.
I was wondering about the limitation of STD.
Yeah. So
the solvent that you're using, and whether you tried some natural product.
Okay. So the most important limitation of STD is its binding. If a molecule is combined in the pocket and don't dissociate, then you will not observe STD effect, because it's like a reversible reaction going on. The one molecule, small molecule, goes into the pocket, get some transient NOE, and it go out from the pocket, but it's a small molecule, so the relaxation is not faster as compared to the protein. So, and in the meantime, the other small molecules are also going and coming back. So the higher the concentration we are using in the ligand, that's why we got good signal to noise ratio. And if the ligand is very much tightly binded or very loosely binded, so we don't observe STD effect. This is the main limitation. It just have usually shows some micromolar to millimolar KD of dissociation constant with protein and ligand. But if it is less than or higher than that, we don't observe STD effect. And second limitation, the small molecules are mainly soluble in organic solvent, and proteins are soluble in buffer. So this is a big challenge for so we what we do, we can, DMSO, deuterated DMSO we use because it's, but it's a very minimal concentration we want to use so that the protein conformation will not be disrupted. Yeah, these are the two important limitation of STD NMR.
I have another question. When you do the mixture of promising compounds with the protein, sometimes when you have a mixture, you have what we call it like synergism or something like. Can you observe such cooperative behavior when you have a mixture?
Yeah, I explained you in my lecture as well that that when we are doing mixture, we have to compromise. Yeah, there might be some interaction that we missed because these molecules are also interacting with each other. So so there is some compromise, but just to save our NMR time, we just want to, this is the compromise in STD of the mixture that we are doing.
But if you have larger effect because you have two compounds together.
I'm sorry?
If you have larger effects, you have two compounds.
Yeah.
They are doing their interaction with the protein when they are together, but when they are alone, they are not.
Yeah, that we have not observed. Yeah. With some limited observations so far, experiments that we have done, this we did not observe.
We have one more question. Short follow-up question. How much do you use?
Maximum we are using 5 microliter in 5.5 microliter in 550 microliter samples. This is the maximum that we are using.
And just a short comment. I like your discussion. The docking is in vacuum. Life is important. The experiment is important.
Thank you, Professor. Yes, Dr. Sami.
Thank you very much for this informative lecture. Just a small comment. You said in your presentation, thank you very much. You said in your presentation you are using a relaxation delay like 10 seconds.
Yeah.
It doesn't make the experiment very long.
Yeah. It makes.
So you need this very long relaxation.
Yeah. We want, because before giving the next pulse, we want our system has to be relaxed back completely. We don't want any artifact. That's why. Usually Bruker recommend maximum 5 seconds. But I have discussed with Bruker. So because I wanted to change this, and I have a discussion with them, and they also suggest you can increase it. So with different relaxations time we have done, and we optimize like 10 seconds.
So usually, I mean, one simple experiment, how long does it take? The whole experiment?
For one STD with the 512 scan, usually takes 6 hours, 6 to 6.5 hours. Yeah. Yeah.
Thank you for the presentation. I just want to comment on a question about protein-protein interaction. So there is in bioinformatics, there is a tool for checking or prediction any protein with another protein. So you can upload your structures, compounds, and predict there is a possible interaction before you can go to your experiment.
I couldn't get your question.
I want to comment on her question about the protein-protein interaction.
Protein-protein interaction?
Yes.
Okay, we haven't talked the protein.
Yes. There's an interaction between the two proteins or the compounds before you can go to the mixture. So you can get a clue or a possible interference between the compounds.
As I understand, you are talking about if the same, because there are several protein molecules and we are irradiating the protein molecule and these proteins are interacting with each other. Okay, so yeah, these are the in a system there are many molecules, like we calculate using Avogadro number. But when we saturate a particular chemical shift or a proton at a particular chemical shift, so all the protons at this point, because we lock in the NMR, the spectrum is the peaks they are coming at the same position because we lock it using different deuterated solvents. So they are coming at the same point. So when we irradiate one, it doesn't mean that we are irradiating one protein molecule. It means that particular proton, which is present in all protein molecule in the system. So that we are irradiating. And definitely it transfer through spin diffusion to all immediately. The bigger the molecule, they have lots of pathway to, the pathway that is allowed and which is not allowed in NOE is just transfer, and also cozy relaxation, lots of different types of relaxation. The bigger the molecule, immediately relax by try to relax quickly. So further, if you want to see the different proteins interaction, there are two different proteins that you can do, but not using STD NMR. You have to use 15 and labeled. One protein is labeled, the other is not labeled. Yes, sir.
Actually, here we are actually looking at one kind of protein, it's a particular enzyme. And we are looking at its interaction with a particular ligand. So step one, you irradiate that part of the protein to that region where you think the active site may be, strong irradiation. Step two, spin diffusion, it spreads across that enzyme. Step three, the ligand comes in and into the cavity, the active site, and through a nuclear overhauser OE effect, nuclear overhauser enhancement effect, there is a transfer to the ligand, and you then do a difference spectroscopy. So with or without that ligand there, and then you are looking at a very simple NMR of just the ligand with the enhancement of the signals shown. So you are just looking at where the ligand may have many different groups in it, but those which are closest to the active site will be greatly enhanced. Those which are farther from the active site are less enhanced. So you can this gives you an idea of which way the ligand is entering the active site and which are closest to the active site of the ligand. But finally, you are getting a simple NMR, which is a difference NMR, showing only the enhanced protons of the ligand. Now, if there are other proteins, here you are looking at an enzyme. Of course, all enzymes of the same type, the same experience in the same way. But if there are different proteins, then you can have, of course, complications. But here, the basis of this experiment is that you've got a pretty purified sample with one kind of protein or one kind of enzyme. So there are other complications are not taking place. I hope I'm clear. Thank you.
Thank you so much, sir.
I think we have time for the next presentation.
Thank you. Thank you. Thank you, sir.
Your presentation is already there. Is it open? What slide? What's the slide? Selector. سامح بس. Okay.
Assalamu alaikum. Our next speaker is Dr. Sameh Abu Zaid. Dr. Sameh Abu Zaid got his BSc from Department of Pharmacy, and he currently serves as head of Department of Pharmacognosy. He obtained his PhD from Tokyo University. He was a postdoctoral researcher at International Center for Clinical and Biological Sciences under the supervision of Professor Dr. Muhammad Iqbal Chaudhary. He is working as university staff member since March 2005. He was promoted to professorship in December 2016. He was a visiting scholar at the laboratory of Professor Guido Pauli, UIC, in 2014. He supervised 25 master and doctoral students working. Also, he works in isolation and structure elucidation and testing biological activity of a plant, natural products, and production of medicinal plants using plant tissue culture and hydroponic techniques. Please, you are the driver now.
Thank you very much, Chair. Let me first thank COMSTECH, and it's a great pleasure to be with to meet Professor Ata Rahman and Professor Chaudhary. Their contribution actually to the many chemical and biological science is outstanding, and I'm very pleased and very proud to be one of the affiliated to the International Center for Chemical and Biological Science. Also, I have to thank Dr. Amal and her magnificent group for the excellent organization and for the hospitality. يعني شيء مش غريب على الشقيقة الأردن. الحقيقة بشكر حضراتكم جدا على الاستضافة وعلى الكرم الضيافة. متشكر جدا. Let me begin this presentation. We will talk about another magnificent aspect of nuclear magnetic resonance spectroscopy, which is a quantitative aspect of this technique and how we use it in natural product chemistry. We are going to cover these points. First, we give a simple introduction to the technique, and then we talk about the two mostly used methods, relative and absolute. Talk about the experimental parameters affecting the QNMR, and give some application from our work for the use in natural product analysis, like flavonolignans, like the products in the market, quality control for the products in the market, and determination of the fatty acid composition in the fixed oil. So, first of all, nuclear magnetic resonance, as you all know, it is about the study of nuclear spins for the NMR active nuclei when it is influenced by applied magnetic field. And what we measure here, it's kind of absorption spectroscopy. What we measure here is the absorption of radio frequency wave for applied to this nuclei. Magnificent aspect about NMR is that the signal in NMR, the very simple signal in NMR, is accurately or can be used accurately to determine the concentration of chemicals in solution. So this is the main principle of using this technique for quantitative manner or for measuring the amount of chemicals in solution. The integrated intensity, let's call it here the area under the NMR signal, and call it signal, not peak, is directly proportional to the number of nuclei giving rise to these signals. However, we have to take into account a number of parameters which can affect this technique, which can give accurate and reproducible or precise determination or quantitative determination for the concentration of molecules or we make this technique more accurate and more precise by taking into account some acquisition and processing parameters for the NMR experiments. First, let's compare the usual method for we use for quantitative determination of compounds like LC liquid chromatography coupled with ultraviolet spectroscopy or coupled with mass. In this case, the technique is really very time consuming. We need time to equilibrate the instrument or the equipment, and we need to have some response factor, molar absorptivity in case of ultraviolet spectroscopy, and ionization efficiency in case of mass spectrometry. And of course, we need reference standards, and we need to establish a calibration curve from these standards. On the other hand, the NMR is much faster, doesn't require this compound-specific response, and it can be done with some techniques without reference compound and without having a calibration curve. What nuclei here we can detect? Almost all the NMR active nuclei can be detected, like proton, carbon, nitrogen, 15, fluorine, and silicon, and phosphorus. But we usually use, or the most commonly used, is the proton NMR, which is more sensitive, of course, because of the high relative abundance, and of course also because of the more favorable T1 relaxation times than other nuclei. Let me give you at the beginning a very simple experiment for a compound called silibene here, kind of antioxidant compound present in silimarine mixture, which is very common drug for treatment of hepato diseases or liver diseases. This is a simple NMR, proton NMR spectrum for this compound. If we change the concentration of the compound, the molar concentration here can goes in a very good, excellent correlation, as this curve shows, with the signal of the compound from low concentration to higher concentration, as you can see on this expanded region of the spectrum. So we have to define here two methods used for NMR. First, the relative method. We compare the integrals of the signals to other signals present in the spectrum. It's very commonly used in organic chemistry, people working in organic senses, and we get the ratio of the compounds in a mixture. Like, for example, if we have two flavones, quercetin and camphorol, very closely related, just camphorol doesn't have this OH group, or I'm sorry, OH group at the ring B. We can easily compare the ratio of quercetin and camphorol from this spectrum and with a relative, giving a relative expression for their quantities. On the other hand, we can also know the actual compound, the absolute compound concentration of the compound, but in this case, we will need a standard. Standard doesn't have to be structure related to the analyte of interest, and we can get, as I said, the absolute concentration as this curve is showing. For example, if we need to evaluate the concentration or measure the concentration of cation, and then we can use a DM2 oxide, and then as a standard, giving a signal away from the signals of the analyte of interest, and then we can make a simple calculation actually for the amount of cation in solution. In the next slide, I can give you, I'll skip these slides, give you a step-by-step explanation for the both methods, the relative method and the absolute method. Here, just like I like to comment on the advantage and disadvantage of the relative method. The advantages of the relative method is a rapid and efficient explanation for the purity of any compound. It can assess actually some impurities representing structure-related or unrelated compounds present, even the solvent, for example, but unfortunately, it doesn't give an idea about the non-observable impurities like silica, like salts during the isolation of compounds, or even water present, which can contribute to the overall sample weight and can give overestimation for its purity. On the other hand, the absolute method, which we use, we need here a calibrant or a calibrating compound. It has to be, it has to contain the nucleus of interest. Its signal hasn't to be overlapping with the analyte signals. It preferably gives a singlet signal. It has to be chemically inert, doesn't interact with our analytes of interest. And of course, soluble in the solvent we used, flow volatility of reasonable relaxation time for the experiment not to take a very long time. It has to exist in a pure form commercially, and it has to be easily weighable. And even some methods are now using the signal of the solvent in which we dissolve our analytes of interest as a calibrant for our analytes. So things are getting much more easier. These are some of the calibrants usually used for as a calibrant for NMR. It's absolute determination of compounds in solution. As you can see, it gives a very simple, spectra, which usually we can choose from them which one which doesn't interact with our compounds. A very simple workflow actually for this technique, which is given by Sigma Aldrich here. We get the sample, and it's and the internal standard. If we use an internal standard here, we'll make an accurate weighing, very essential step in our experiments. And then we dissolve our compounds in the NMR tube. Has to be fully dissolved without any impurity, without any particles, of course. We made our simple NMR measurements, and then we get our spectrum, the FID, and then Fourier transform it to the spectrum in the chemical shift domain. Here we have to define some acquisition parameters to get an accurate and precise, as I said in the beginning, quantitative NMR experiments. We have to define some acquisition parameters and processing parameters for the raw data we get. For the acquisition parameters before doing the experiments, of course, we have to use the decoupled technique not to get the satellite signals and complicate our spectrum. We have to make, preferably to make spinning off, not to get spinning side bands. We want to get a very simple spectra as much as we can for proper quantification. We have to make accurate shimming, and this is also very essential. We have to make homogeneous. We have to make preferably manual shimming to get homogeneous field, which can make accurate determination for our compounds. We have to calculate the relaxation delay and put it into account for most of the protons in our sample, expect them. And then we have to use the proper pulse widths, which gives the most accurate, usually the 90 degrees pulse available in Bruker instruments. And we have to get the proper acquisition time for the getting the FIDs in complete form. The other processing parameters which also have to define, many softwares now are available. We have to use the proper software which can give us the right and proper processing for our spectrum or our raw data. We sometimes use what is called zero filling. We put some imaginary points to increase the digital resolution in our spectra. And we have to make phasing for our spectra, preferably also manually using the software package. Baseline correction is essential, and referencing for the solvent used or for any internal calibrant used. For example, this is a very simple experiment called inversion recovery experiments, in which we invert the signals and then recover it again, and then from this experiment we can calculate the relaxation time of our experiment. This has to be known and have to be taken into consideration for a proper or accurate NMR determination. Then I will move to make it much more live or much more simple. I will move to some of the examples we are using
or have already used in our group for quantitative NMR determination. All of them are in the natural product arena. We used it to study flavonolignans, kind of flavonoids coupled with coniferyl alcohol present in the very well-known antioxidant mixture called silimarin, available in the market worldwide. And also we used the technique to study the products of this silimarin in the market. And we use third, we use the technique to study the fatty acid composition in the fixed oil in general. First, the silimarin or these flavonolignans are the main components in silimarin. This is a fruit extract obtained from some plant called Silipum marianum, quite available here in Jordan and in Egypt as well, many areas in the world. It is used for centuries for treatment of liver diseases, especially before the availability. It was the main drug or the main hepatoprotective agent before the availability of the new anti-HIV, HCV agents. And it's nowadays also it has some other recent reports for its capability as a chemopreventive agent for prostate carcinoma. Inside the fruits, actually this is a fruit of this plant, the mixture called silimarin, which is defined as the standardized extract from the fruits, which is a complex mixture here. It has to contain like from 65, according to most of the pharmacopoeias in the world, 65 to 80% flavonolignans, and its brand name here from the famous German company called Ligalon. Here again, this is the silipene, one of the main component in silimarin. I have shown this slide before. There is a correlation between the signal or the intensity of the signal or the peak area or the signal area and the molar concentration of the compound. So for this silimarin component, which we isolated from Silipum marianum fruits, we can take the QNMR spectra here for some of these compounds actually, applying the acquisition and processing parameters of QNMR to get information about the purity of the compounds which we have isolated. Also, another application, we were trying to develop a new methods for extracting this silimarin mixture, which is a very important raw material in the pharmaceutical industry all over the world, by making a separation between the kernel and the internal part of the seed. We are trying to use these, the kernel and the pericarp. We are trying to use the pericarp here for isolating silimarin or for producing silimarin. And then we used the quantitative NMR method to compare between getting the silimarin from the pericarp, not from the whole fruits, and we patented this method, using also the single 90-degree pulse experiments for 60 seconds relaxation delay, 4 seconds acquisition time, 64 number of scans in a non-spinning mode. For the NMR can tell us it contains much higher or extracting silimarin from the pericarp. By putting this step first, it can give us the spectrum at in the blue color here in the back, can give a silimarin much higher than that we can get from the whole fruits. In addition, also NMR with its capabilities to give us some structure information about the compounds, we looked at our spectrum and then we look back to the other flavonolignanes which may be unknown. Here we found some report which was published in long time ago in 2007 in Planta Medica by some group from New Zealand, talking about one of the compounds which were very unknown called silyamandine. And then the group were expecting this compound or proposing this compound to be formed in an aged extract, an extract which has left in refrigerator maybe for weeks by some decomposition reaction from the well-known silydianine flavonolignan compound using this scheme. So the group in 2007, or this report in 2007, were expecting silyamandine to be formed with like this chromatographic separation, which is very well, which is not very well actually, doesn't show very good separation here. They were expecting silyamandine to be a decomposition product coming from the well-known silydianine. We'll look back for our spectra, getting silymarine seeds or Silybum marianum seeds from different parts in Egypt, and then we analyze them. We find silyamandine, one of its signals, the proper, we actually isolated silyamandine in a pure form, and then we took we measured the NMR spectrum, protein spectra, and then we found some correlation. Here, the correlation map with two compounds, silydianine and isosilylcrystine. Then we look back for how these compounds are biosynthesized in nature inside the plant by simple radical coupling between coniferyl alcohol radical and a taxophylline radical to give here isosilylcrystine out of this intermediate. This intermediate can undergo ketoenol formation and then epoxidation to the silyamandine, and we proposed silyamandine to be a natural compound, not a compound, can be biosynthesized from the same way as, or a busway from, side busway from these isosilylcrystine methods. And this was actually, as I said, supported by the correlation with the isosilylcrystine and silydianine, and at the same time supported by taking the simple experiments, not aged extract experiment by NMR, which can give us the details of the structure determination. Second application actually here is we can also use the QNMR to study or to make quality control for the products available in the market in my country. For example, silymarine is present, it's either registered as drug or as dietary supplements. These are some commercial names by different pharmaceutical companies. These are the products, and it's very important to do quality control for this product. We studied the NMR spectrum for all of these products, and we calculated or measured the flavonolignanes, total flavonolignanes here. We also did the in vitro antioxidant activity against the DPPH, famous radical, and we correlated this actually with some commercial data available from the very well-known IMS, Intercontinental Marketing Service. We here like to do like correlate the amount of the active constituent, its activity, with the annual consumption and the annual sales of these products. We did some chemometric analysis. Here we found two products that are considered to be outliers from the other products. This was Silymarine Plus capsule and Silymarine Plus sachets produced by one pharmaceutical company. Just by correlating the total flavonolignanes with the activity. Interestingly, we found that these two products are really in the space of the vectors of the biological activity and the flavonolignan content, and also these were in the space of annual consumption and annual sales as calculated from the IMS data. And interestingly, these were the most famous and the most sold product in the market from Silimarin. A third and last application here, we also used QNMR to study the fatty acid composition in fixed oil. As you all know, fatty acid is mainly composed of a glycerol molecule esterified to a fatty acids, and these fatty acids can be either saturated or unsaturated, like oleic, linoleic, or linolenic acids in most of the fixed oils we know. A very simple and very nice, actually, NMR, proton NMR spectrum can be produced out of this fixed oil in a very easy way and short time. And this spectrum, if it can be well studied, each of these signals can express protons in the fatty acids or in the glycerol molecule. By simple calculation, we can get, by using these protons, like for example, here, linolenic acids containing a terminal methyl group. This is the signal for these terminal methyl groups. So measuring the content of linolenic acid can be easily estimated by measuring the area of these signals. And then we go to the next signals and we subtract the other common signals with other acids until we get the content of linolenic, linoleic, oleic, and other saturated fatty acids. A very simple experiment which was reported actually long before in 2010 by Anderson and co-workers. We use this method to calculate or to study actually the composition of the fixed oil obtained from the kernel of the seeds of this plant. We get our NMR experiment the same as the last slide, and then we calculated the content almost doesn't contain linolenic acid or undetectable here, linoleic, oleic, and saturated. In total, we got a little bit higher than 100%, and this can be attributed either to other free fatty acid present in the fixed oil, and this can be detected easily by a singlet around 11.5 ppm, or by presence of glycerol esterified to only one mono or two diacyl or two fatty acids. And this actually was the reason here, by detecting a doublet of doublet signals by enlargement of the spectrum, in the enlargement of the spectrum around the glycerol signals. In short, this technique is simple, rapid, and easy to implement. It is specific. It doesn't require reference compound for quantification. Cheap internal standards commercially available can be used, while maybe some of the drawbacks here, of course, it needs the availability of the NMR, nuclear magnetic resonance instrument or equipment, and of course, the low sensitivity compared to other spectroscopic methods like mass spectrometry, for example, is still a problem. The quantitative NMR method can be integrated into the routine qualitative method which we all use for structural elucidation, and it can provide a value-added dimension to the standard spectroscopic techniques in structural analysis when analyzing the natural products. Finally, I have to acknowledge some of this work has been done in the University of Illinois with the Professor Colguido group, and some of the work, especially the fatty acids and the quality control for Silimarin products, in Bani Suef University in my country. Now I'm working for Heliopolis University for Sustainable Development. Part of the work also is supported from the Science and Technology Development Development Fund in Egypt. At the end, I'd like to invite all of you actually to participate or to follow us in this year in September in our natural product research network called NAPRECA. I have my friend here, Dr. Solomon, is an integral part of the important part of this network, which can combine almost many, like 20 or 22 African countries working in natural product chemistry. We have a biannual conference since the 8th of the last century. This year, the conference will be in Zimbabwe from 18th to 20th of September. And it covers many topics in natural product chemistry. Hope to see all of you there. Thank you very much.
We have like 10 minutes for questions. Anybody has a question? Hi. Dr. Ahmed.
Thank you a lot and welcome to Amman. We missed you yesterday.
It's my pleasure actually to be in this lovely country.
Most welcome. And thank you for the nice lecture. I was wondering, when we heard from Atiya, she was talking about using the NMR, the time of the NMR, the cost of the NMR, and using sometimes mixture to reduce the cost. And I was wondering about the NMR, quantitative NMR versus simple UV, for example, simple FTIR, which is much easier sometimes and for industry because you're using it for industry. And for research, sometimes you go for more costs, but for industry, for application. So what do you say about that?
Well, I have to tell you that we have in our countries, actually, in our scientific communities, to be ready for this technique, because if it's not already appeared in the famous pharmacopoeias in the world, the USP, United States Pharmacopoeia, the Japanese Pharmacopoeia, and the Chinese Pharmacopoeia. So it's beginning to get its strengths, and it's beginning to get its application in analysis of many drugs, not only natural and synthetic as well. Getting the capability of combining both qualitative and quantitative, doing qualitative and qualitative experiment at the same time. You know, we can get qualitative information, and at the same time, if we look for some of these acquisition and processing parameters, we can get also the quantitative determination. But we have to put into account that it is becoming, or it has already become, an integral part of the famous pharmacopoeias in the world. We still, I think, in Egypt, I'm talking about in my country, we still don't have any pharmacopoeial methods using quantitative NMR for analysis of drugs, like a monograph in the Egyptian pharmacopoeia, for example. But it's already appeared in USP, Japanese pharmacopoeia, and Chinese pharmacopoeia. Yes.
And also that advantage will be used for mixtures.
Exactly. Yes. Exactly. Yes.
Just a small comment. The NMR is now one of the tests that must be done according to FDA for all the drugs, even in industry. So I think a pharmaceutical company, they have to start buying or doing it, because now it is a must, not something complementary. Also, NMR, you can analyze mixtures at the same time. Exactly. And actually you can identify them if you do the NMR without separating them.
For quality control, yes, yeah. Exactly.
Thank you, Doctor, for this nice presentation. I was looking forward, honestly, to see how can I use this method, which is, first of all, it's simple and quick if you have the machine, which is so expensive.
Yes.
Buy so expensive drug as well.
Yes.
And you have to have an experienced personnel
Yes.
and use reputable data. However, if I'm working in a natural product research, how can I make use of this method to at least highlight the presence of certain important components in an extract? What you have shown is either a mixture of a known composition, first of all, and then you have to, okay, maybe you can run it without the standard, but at least you will have a library for the NMR of the proposed compounds content. But in an extract which contains hundreds, thousands of compounds, how can I make use of such a method? It's going to be so complicated if I just give you an extract. How much you dilute it? You're not going to have pure or at least a clean NMR spectrum. Did you try this, or just it is just for the quality control of certain products? Because I know it's been used for survival.
I can tell you this is really one of the limitations of this technique. You know, it's like the dispersion of signals in a very short spectrum. Dispersion of signal, one of the limitations of this technique. But still, with proper experimental design for the technique, with proper knowledge of what compounds I may get. I mean, before doing the NMR experiments, what compounds I'm expecting, what compound I'm extracting. With proper experimental design, it can be a very useful method for structural elucidation and quantification as well. But of course, as you said, it has some limitations in especially with extract compounds.
Is there a library available for the NMR data of natural products? Because the product has to be almost pure, semi-pure, or just containing a well-defined group of compounds.
But as you know, there is a small differences according to the solvent used, according to the strength of the instrument used, either 400 or 600 or higher. Yes. So, to my knowledge, I mean,
it's not like the UPSC
or the mass. Yes.
We have library. You can just match.
To my knowledge, still we don't have this library for NMR that's dedicated to NMR. Yes.
Because you mentioned that USP now is
certain monographs. USP, yes, containing the method itself, or how can we measure in the monographs. Yes.
Thank you.
I'd like to ask you about the use of slimarin containing products, just to share information what happened in my country. Because from my reading, FDA has not approved slimarin for health supplement use. In Malaysia, it is categorized as low GLIP. Low GLIP means just for general use, for health, or maybe for liver tonic. You cannot put any clears for treatment. You mentioned, I'm not sure whether in Egypt you can use slimarin containing product for treatment. Because so many diseases, liver diseases. Although you say in the pharmacopoeia, not sure what's happening in EU, European Union country. They in fact prescribe standardized several products for patients. But in Malaysia, none of the herbal products being categorized or being accepted for treatment. For that, because there's no complete clinical trial data. What they have is preliminary, but that's not enough yet to classify even standardized herbs, except for those commercial herbs, those being used or being dispensed in pharmacy with clinical trial data. Then can you question what you do from your presentation mentioning there's a lot in the market, as drugs and also as dietary supplement situation.
Actually, this is not only in Egypt. This, as you said, in also some of the European countries, the same silimarine with the same dosage form, with the same quantity, but it is registered by the companies either as drug or as dietary supplements. I can tell you before emergence of the new treatments for hepatitis C viral infections, silymarin was the main and the only agent which can most of the physicians use for the patients as a hepatoprotective, not as treatment, of course, but against the virus, but to protect the liver cells, which can still work till 10% of the liver cells are alive for the HCV or hepatitis C viral patients. Thank you.
Do you need to use a microscope for such analysis?
No, the proper, the simple book. Yes, the ordinary one is used.
Such a small amount.
Yes. Depends on the level. It depends on the level of the measurement you are using. But the ordinary probe is very, very much used in many experiments described in many literature data.
Thank you. We do not have more questions. Let's thank Dr. Sam one more time.
Thank you very much. Thank you.
And now we have 20 minutes for coffee, then we will return back.
Thank you. So the next session is on practical, is a practical session on NMR, and it is going to be presented by Professor Dr. Atiyah, Professor Dr. Amal Al-Abodi, and Professor Dr. Jalal Sera. And it's going to be two sessions, one after other. So, thank you.
Assalamu alaikum. Just let me first say how we are going to run this session. This is a practical session oriented basically to the students, to just to show them how can we solve the structure from NMR. So Professor Atiyatul Wahab will give you a short introduction, just a very short basic introduction. But I would like first to ask the students who did not attend any course in NMR, just raise your hand. So any course, you don't know anything about NMR. So all of you, you have some idea about NMR. So we can just have a very short just introduction. And we have distributed some spectra in which we will be sharing them and discussing how we are going to solve to reach the structure. During this time, we have many experts here in NMR who may volunteer to help the students. Basically, I'm here, Dr. Jalal is here, Professor Adar Rahman, Dr. Salim Sabri, Dr. Musa, we have many experts also from Egypt, Dr. Sameh. So we have many experts in NMR, also our colleagues here. So the students, whenever you have any question, you can ask, and we will be coming to the table to help you, to show you directly how to reach the structure. So I'll give the floor now to Dr. Atiyah, please.
Assalamu alaikum. So we'll be starting interpretation section. Actually, it's I hope it should be clear. because the handout which is given, it's very tiny and can't be read. So, but you have this one in your. Can you hear me now? No.
Yes.
Okay, yeah. So this section, as Dr. Amal explained, is basically for students, and I request all the NMR experts. There are many experts, including our teacher, Professor Taur Rahman. It's difficult to speak in front of him, but I'll try again. So what we are supposed to do using NMR spectroscopy is to elucidate the structure. But since we have short of time, so a structure is in front of you. Okay, so what we are going to discuss is assignment and how to read the spectrum.
Maybe this one.
Okay.
Talk.
Yeah, thank you. So, okay, first, the basic, there are different steps that we have to take care if we are talking about this. Okay, so there are several steps that as organic chemist, as a structural chemist, that we have to take care of it. We don't want to take our samples directly whenever we purify or synthesize any compound. There are several steps that we need to take care, because we don't want to waste our institutional resources for something which is not very important for us. So the first step, whenever you have a pure compound with you, either you synthesize, isolate, biotransform, in any way, you need to check the mass of that compound, and then HR, high resolution. So with high resolution, can match with the library, existing library, and get some idea of the molecular formula. And then the if your compound is pure enough and you have some good quantity, with the new electronics, any molecule with the small molecule with 1 milligram of quantity, you can use for proton NMR spectrum. For C13, it require if you are using like 1 milligram, it requires much longer time, because it's usually detect on the natural abundance, and C13 is more than 6,000 times less sensitive as compared to proton NMR. So we record the proton NMR spectrum in the first case and try to match with the literature, with the mass and proton NMR. You have to try and match with the literature, whatever existing literature of your compound or the plant or metabolite from where you have isolated. And if something exciting, you can proceed further. And if something is already reported, then you don't want to waste your institutional resources for recording 2D, NMR, and C13, etc., because it requires more time, more NMR time to be recorded. So this is the first step, that you have to always take care that every step you need to go back, record one spectrum, check with the literature. if something is really worth proceeding further, or otherwise you don't want to waste your time also. So in this case that we have the mass, which is 228, and we can check the fragmentation pattern as well, and to find out if the fragmentation pattern is logical or not. And then this is the proton NMR spectrum. There are some important information that you always need to check. First is the frequency of the spectrometer on which it is recorded is SFO1. It's always like it is recorded on 400 megahertz. So whatever the frequency of the spectrometer, it's recorded, because this information is important for you when you want to calculate the J values, because you must know what spectrometer this is being recorded. And second, this is the number of scan. NS is the number of scan, which is 32, in which it is recorded. The solvent it was used is deuterated DMSO. So we use deuterated solvents because first we have to lock our signal, and we don't want, because solvent is like several fold more as compared your compound. Because if we have like 5 mm probe of NMR spectrometer, which is usually the case, then you have to use 550 microliter of the sample of solvent. So the solvent and if your molecule is like 1 milligram or less than 1 milligram or 2 milligram, so the solvent is several fold more as compared to the molecule or that you are going to analyze. So we don't want to see the signal of solvent. So that's why we use deuterated solvent, and deuterated solvents are very expensive. The most cheapest one in deuterated solvent is D2O, and then no, first is chloroform, then D2O. You know how many deuterium is there? The more the deuterium, the solvent is more and more expensive. So CdCl3 has only 1 deuterium, so it's the cheapest one, and the more deuterium is present in the solvent, it's more expensive. So use the solubility, and that's important thing that I personally have observed, that many students they are not sure which solvent their compound is soluble in, and they just submit their sample for the NMR analysis, and often it happens that the sample, and if you are recording a solution NMR, the sample has to be soluble. If it is not soluble, you are not going to observe anything. So if you are using an NMR, is not the technique to check the solubility. And that I personally have observed that many students do such kind of things, like they are not sure which solvent their compound is soluble. So they just randomly write and find out that this is not soluble, then such an NMR cannot be done. So the solubility need to be checked. If you're using solution NMR, your compound, your molecule need to be completely soluble, so that you can run the spectrum NMR, and you can get the good signal to noise ratio. And here the number of scan was 32, and Dr. Iqbal will explain you in his lecture. This is the simplest one with proton NMR with 90-degree one pulse. With the 90-degree it's given. So NS is like number of scan is like, here is the number of times of experiments that you are recording one after the other two, just to get the good signal to noise ratio. And it's so one after the other. The 32 NS 32 means 32 times this experiment was recorded. So and here the D1 is the relaxation, that you can see the 2 seconds. Usually in Bruker, 1 to 5 seconds is the recommended based on the type of the experiments that you are doing. So, and the temperature you can definitely check, and solubility of each molecule also depend on the temperature. So you have to be careful that you have used, sorry.
Can we just give it a break short, break a little bit so that the other trainers can go around the tables, and repeat a little bit more practically what you have said, because they were talking and looking for the pages. Is this possible?
Yeah,
we will give you a short break. Yeah. So the other trainers will go around and make sure that they are following to the same pace.
Yeah.
Good.
Yeah, sure. I'm just on this page and I'm trying to explain a little bit more detail. So I'm not changing the page right now. So there are further details that's before experiments.
But I have. Hello, just one question I'm going to ask Atiya. Maybe it's going to be really useful for you. Can you give me some attention, please? The solubility, she actually addressed this problem. My question to Atiya, when we do solubility tests for our sample, of course we will do it in undeturated solvents because deuterated solvents are very, very expensive. Can there be a difference in solubility between solvents, normal solvents, and deuterated solvents?
I personally never find such kind of difference. Yeah.
So, good thing is you do your solubility test in normal solvents, check the suitable one and report it. There will be very, very small differences between the solubilities in normal solvents and deuterated solvents. You have
So one important thing that we also observe.
This is better now.
Yeah.
There's a lot of expert
Yeah.
people and they can discuss. And each one can discuss with their students. When it is not good,
Yeah. Yeah.
and it is better to discuss.
Yes. So should I? Yes. Should I stand here?
You don't have to share. You can share any of these sessions if you would like.
Yeah. Yeah.
هذا المفروض يشتغل معك صح؟ مش عارف. لا مش عارف. هذا لا سبورت البي دي اف. هذا لا سبورت الباوربوينت. بس اذا بدك خلي الليزر عندك، في الليزر معكم كبوينتر طلع بوينتر خلي معك الطاوله حددوا لي ايش من خلي ديسكس عليه الاحترام التام.
Like, if you stand here, without the mic, then you can discuss with them whatever. You can close it. Otherwise, nobody is hearing me. I'm talking now, but nobody is hearing me from the room. But are you? I don't know whether the people online are hearing us. How can we know? Let me send the person responsible for the IT or what? Yeah, let me ask Shamshad. Yes, and I'll send you a person responsible for the IT so that you can handle the people online. Yeah, and you share the screen. You discuss with them.
Hello, can you Shamshad ki awaaz aa rahi hai? Yeah.
Yes, ma'am, Ari. Yes, ma'am. Yes, ma'am, I can hear you.
I'm person responsible. Okay.
Shamshad awaaz aa rahi hai?
Yes, ma'am.
Okay. So I'm here for the online participants. There is actually different groups which has been made here for those participants who are present physically. So I'm just explaining things for the online participants. So let me go back to this spectrum. So I will explain briefly that this is the structure of the molecule that you can observe, and this structure needs using an NMR spectroscopy. We try to solve the structure of unknown compounds, but here, since the short of time, we are just going to know the interpretation of this. So here you can see what we are expecting to observe. We should observe, so this is protein proton NMR spectrum. We can observe this metal and this methoxy group, these protons, and there is 1, 2, 3, 4, 5 aromatic protons, and these are the 2 methylene groups that we are going to observe. So what we expect so this is the proton NMR spectrum, and it is recorded in so in the first instance, here you can see that this is the, we are using deuterated DMS, but there are, if given that deuterated DMS is 99.99% deuterated, but there are like 0.001% hydrogen. So the residual peak we are going to observe here, and this is the moisture content DMSO. So that we are going. So this is a beauty of NMR spectroscopy that we can observe on the a particular type of the nuclei based on its resonance frequency that Dr. Iqbal will be discussing in his lecture. So here, as we have seen that there are 2 types of metals, one with the OCH3 and one with the a metal which is attached to the carbonyl carbon, and there are 2 methylene groups. So the methoxy, which is present on the aromatic ring, usually appear between 3.8 to 3.9 ppm. Here you can see 3 proton signal, and it can be regarded as a methoxy proton. And in the proton NMR spectrum, usually whenever we are writing the paper, we have to write like 2 points after 2 or 3 points after the point value. So like we can't say like 3.8, but we will say like 3.84, 1 or 3.8. So similarly here, 2.09 is the metal signal that we are observing. And these are the 4 protons. Here, these are the integral value that you can see, and these are the 4 proton signal which are splitted because these protons are correlating with each other. Yeah.
all is well? خلاص ما في. You don't need any technical assistance? Okay, thank you. So these are the aromatic signals that we are going to observe in the structure. We've seen that 1, 2, 3, 4, 5, 6. 6 protons that we should observe. Okay, so here we can see that this proton should have splitted with the ortho coupling of this, and this we are going to observe the ortho and meta coupling of this proton. And here this should be a singlet, and these are the 2 doublets that we are going to observe.
Can you put the aromatic region for this one?
I'm discussing online with the online participants.
Okay, I just want they want to see the aromatic region enlarged. Yeah, okay. Thank you.
So here you can observe these different type of proton that you can see that here these are the 1, 1, 1 proton degradation, and these are the 2 protons. This is doublet and this is also doublet, but it's not a split. And so it's kind of broad. And here we can calculate the J value because this spectrum was recorded on 400 megahertz. So if you would like to calculate the J value, what you need to do, for example, if you want to calculate the J value of this one, so we subtract 7.248 from 7.252, and whatever the value we get after subtraction, we will multiply with the 400, which is a spectrometer frequency on which the spectrum is recorded. So we will get the value in hertz. Since the splitting is very small, so we calculate in hertz. So this is the way in which the ppm can be converted into hertz. So if you would like to have the real frequency, because we are using this ppm usually, but if you want to see the real value in hertz, so what you can do, you can multiply the ppm value with the spectrometer frequency, then you can get the value in hertz, because the spectrometer in megahertz, and this is ppm parts per million. So then you can get this value. And so these are why you want to use? Oh yes, we can. But I'm discussing with online participants. Yeah, I'm discussing with online participants. Okay, so not here.
To point to the screen.
No.
Can you just freezing the screen?
No, because I'm discussing with online participants, this computer. There are many online participants, and I'm using this one for online. So don't consider this, okay? I'm sorry for the disturbance, and actually this one is there are different sessions, groups are going on. So so there are 1, 2, 3, 4, 5, 6, 6 protons there. If you go back to the structure, and these are the aromatic protons. So here we can see 1, 2, 3, 4, 5, 6. And these are number of protons that we observe. There are 2 clean groups we observe. There are 2 methyl we observe in proton NMR spectrum. Okay, let's come to the this is the UV spectrum and IR spectrum. So in IR, the most characteristic, the carbon signal is there 1702, which is the ketone, showing the ketone moiety, and they are very specific. They are fingerprinting region. So specifically finding out this specific functional group like amide, carbonyl, NH, and carboxylic acid, hydrogen bonding, all these kinds, very important. We can get it easily from IR spectrum. And so here there are 3 types of the carbon-13 spectrum. This is the broadband BB. Okay, so this one is recorded 150 megahertz. 150 megahertz means it's recorded on 600 megahertz spectrometer, because you need to divide the frequency with by 4, because 4 times less. Actually, the 600 times
but the resonance frequency of carbon is on this spectrometer is 150 megahertz. So here, how many will go here? Back to the structure, and we'll see how many carbons are there. So we can calculate like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 16 carbons are there. Okay, 16 out of 16 carbon, there are 2 metals and there are 2 methynes, and these are methine. 1, 2, 3, 4, 5, 6, 6 methine, 2 methyl, 2 methylene, 6 methine, and the quaternary. Here is 1 quaternary, 2, 3, and 4 quaternary. So 4 quaternary carbons. So this all in broadband spectrum, we are going to observe all kind of carbons. And here you can see the most downfield signal 207 is carbon and carbon. The most downfield one is usually carbon and carbon. And there are different carbon and carbons, as like this one is a ketone, so it's, they are to be ester, and it's usually resonant from ester, aldehyde. There are different functional groups of the carbon and carbon that you can observe around 180 to 200 210 ppm. So, and then you can observe. This is again, it was, we need to see the solvent. So the solvent here is DMSO. So deuterated DMSO we have used. You can see the solvent here. And here it's, we are recording this C13. So you can see the number of scan here. So the number of scan NS is 15, because the sample should be very concentrated, like 1567 number of scans, and when it means that the sample was really concentrated. But otherwise, is usually if you have like 2 to 3, 4 milligram of sample, it requires like 20,000 scan or 25,000 scan. It takes the whole night to run this experiment. But if the concentration is very good, so the experiment should be less. So here you can observe the 39 point, all these very other the residual C13, which is coupling of proton. So this is this value of you are observing here, because there are some protons, 99.99% are deuterium, but there are some hydrogen remaining. So this hydrogen of the solvent split to make the splitting of the signal that we are going to observe. And here you can see the so here you can see this is there are 2 methylene and 1 methyl and then 1 methoxy. So this could be the methylene, and here we cannot integrate how many carbons are there. We cannot do it because of the different relaxation phenomena. And here, this is the methoxy carbon. This should be the metal, which is attached to the carbonyl, and these are the methylene. But we can check it more carefully by using the depth 135 spectrum. So depth 135 spectrum shows that this is the methoxy, because those which are CH2s, they are always heading downward. So they are one peak 29.0 and the other is 44.1. They are heading downward. So these are the 2 methylene, and this is 29.1 is the methyl, which is attached to the carbonyl, and this is the methoxy. And then you have signals of 6 methine carbon. So here 1, 2, 3, 4, 5, 6. So here this is the beauty, because the scale is bigger from 0 to 10 ppm. So you hardly see any overlap. So like it's not like the proton NMR spectrum that has many overlap, but here you don't observe any kind of overlap, so you can see peaks very clearly. And this is depth 135 and this is depth 90. You can see, and this only shows the methyl carbon. So when you subtract these depth 90 and depth 135 spectrum from whole body spectrum, then you can find out the those carbon which are quaternary carbon that were 4, and this one was the carbon carbon. So there are 3 remaining which are the quaternary carbon. So I would think that the next important spectrum is HSQC. So now you know the carbon values and you know the proton NMR chemical shift. So the next one is the HSQC spectrum. So here you can see the coupling of carbon with proton, which is directly attached. So here you can see these are the 2 methylene groups. We can see it has the carbon value around 29, and this is around 41, 42, and these are the proton signal of these 2 methylene group, and this are the proton NMR signal of the metal, which shows the attachment. And here there is this one is the methoxy group that you can observe, and this is the solvent signal, and this is mixture of the solvent. And here these are the protons and carbon attachment, which is directly correlation. And this protons, if you draw a horizontal line and a vertical line, you can see directly that this proton, which is resonating at 7.1, is actually linked with 119 carbon. So in this way, we observe this HSQC spectrum, and the HNBC spectrum is we can observe like long range correlation. So here, not the direct correlation of proton with the carbon attached, but the 2 or 3 or 4, usually 2 to 3 bond. Sometimes very rarely we can observe the up to 4 bond correlation. So here it shows that this bond is connecting with the carbon and carbon, and the another methyl group which is also connecting the carbonyl, and these are the carbons if you need to draw the line from top to bottom, and these are the chemical shift of carbon. So this is how we read the spectrum. And then the second important one is the COSI. And COSI, there are different type of Toxi experiments. So Toxi is important when you have the complete whole spin system. When you don't have a whole spin system, then Toxi is not so important. And Nosy is important when you want to see some kind of in space coupling. So here specifically where you need to assign the Toxi group, it's important to play, and it's important to see the nosy interaction with Toxi. So here you can see that this is the methoxy proton, and this shows interaction with the proton at 7.3. So which means that this methoxy is showing the interaction with either of these protons. So that's how you assign the presence of nosy. So, or stereochemistry is important if you want to assign the stereochemistry. And the COSI is important if we see Toxi 60. Yeah, it's long range, but we don't have COSI here. But the Toxi are for the whole spin system. So if we can see this are connecting with each other, but the COSI is very important, which shows the interaction of adjacent of the same protons at the same carbon of the adjacent. So right now we don't have, but proxy in that case, in this case, is not so important because it's as important if you want to see the whole spin system. But there is no such kind of a whole spin system. There are two, they should couple with each other. These two should couple with each other. These two should couple with each other. So not the whole spin system, like the sugar is the X system. So that's how we interrelate and interpret the spectra. So rest of the spectrum will be shared to you by the organizer. And if you have any question, please feel free to ask. So we will have a break then afterwards, and then we'll come back with the lecture of Professor Iqbal Chaudhry. So if you have any questions, you can ask me now. So if not, then we'll be back around 2 o'clock with the lecture of Dr. Iqbal Chaudhry that will be discussing about the basic concept of NMR spectroscopy. And hope you guys understand. But if you have any questions, further question, you can send question to the chat group, and we'll try to answer afterwards. Thank you.
So we'll start in a short while.
Hello.
Okay. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم.
I'm trying from here.
On your computer.
Dr. Muhammad Iqbal Chaudhary. I'm impressed, and I will say, as we say in Arabic, وإن صخرا لتأتم الهداة به كأنه علم في رأسه نار. So I want to translate. This to English. Dr. Muhammad Iqbal is like a light on a mountain, so the eye cannot lose him. So everybody see him as a light on the mountain. Dr. Muhammad Iqbal Chaudhary is a distinguished national meritorious director at the International Center for Chemical and Biological Sciences, coordinator general COMSTECH, vice president COAS, and UNESCO chair in natural product chemistry. He is among the most prominent scientists of Pakistan, recognized for his original contribution in the fields of natural products and bioorganic chemistry. He has written and edited 90 books, most of which have been published in USA and Europe. He has supervised 98 PhD scholars for their doctoral dissertation. He is also authored over for more than 1,200 research papers and 40 chapters in top international science journals of the West, as well as 65 in US and international. He has been among the most cited scientists of Pakistan, with citation exceeding 36,000 with H index 80. He is the volume editor of many international book series and journals. So, you are welcome.
Assalamu alaikum wa rahmatullahi wa barakatuh. Can you hear me? It's all right?
Actually, no. Maybe other one is better.
Thank you very much, the chairperson of the session, for his very generous remarks. I think that I deserve them. Okay, so this is the second day of this workshop, and many of you are already exposed to very high quality spectroscopy techniques that you have done from some of the most experienced individuals. my work after this practical session was supposed to be reverse. You know, I was supposed to give this lecture first and to have this practical session for you later. But since I was basically responding on another official engagement, let me just without wasting any time, I'm going to talk about the concepts of concepts, you know, applications you already know. You have gone through the applications of NMR. You have seen that various NMR techniques can be used for a variety of different purposes. I would actually ask you and take you to the most fundamental questions in NMR spectroscopy. And don't laugh at me because there are some very experienced people sitting here and they know this field very well, but for the sake of completion and for the sake of young people, let me take the risk of repeating them. So this is a lecture which I dedicate to my teacher, Professor Dr. Ataur Rahman, and with a very interesting story. Back in 1983, when I joined him, a fresh student who came from University of Karachi, I used to have hairs as long as this. I used to wear jeans all the time, which I often, you know, once a year I used to wash. But I used to take shower every day. And then, you know, second month of me joining Professor Ataur Rahman as a student, he met me in the corridor, he touched my shoulder gently and said, next Thursday, and this is a real story, next Thursday, you need to deliver a lecture on, listen, incredible natural abundance double quantum transfer experiments. He said, what is this? Completely blown apart, inadequate. He said, on Thursday, you need to deliver a lecture for the other students on a new topic, which was emerging called incredible natural abundance double quantum transfer experiment in a liquid. And for a few minutes, I was shocked. I was completely shocked, but then I went to the library and I started looking for it. And then I found the most recent paper by Gunther. And he developed a technique called inadequate, credible, natural, abundant, double quantum transfer experiment. Inadequate. And this was the first time someone from the animal community developed experiments which could identify direct coupling between carbon and carbon. You see, what else you need? We have been working on this piece spectra, trying to figure out proton connectivity, proton-carbon connectivity, and then indirectly carbon-carbon connectivity. The fact is that organic chemicals are constructed, biochemicals as well, based on carbon skeleton. So if you can get carbon skeleton directly, then you don't need us. So I delivered this lecture. I don't know what I spoke. And then he came and said, I think you're learning it. I'd like to write a book with me. Can you believe it? On the third week of my stay with him, they already offered me to write a book. That is my journey, and I want to acknowledge this lecture to this wonderful person, my teacher. I really need to. Okay, so there are only 373 slides, and I'll be able to finish it by tomorrow evening. Is this okay? Okay, if we came to the sleeping and for dinner. What I would like to do is to give you what is NMR. That is what I'm going to speak. So don't laugh at it because this is what you already know. how it is different from other spectroscopic techniques. NMR is a different animal altogether. His theory, his understanding is altogether very, very different from other spectroscopy. Why radiofrequency absorption requires application of applied magnetic field? Why you need to have applied magnetic field in order to have radiofrequency absorbed and you detect that as NMR signal? And I'm going to ask you a question, why not all atomic nuclei are NMR active? Why not? I'm also going to discuss why NMR is so insensitive. It's one of the most insensitive techniques in the spectroscopy. So many factors, magnetic direct ratio, natural abundance, low correlation, different energy states, cortical versus dipole nuclei. So many factors which are damping, which make NMR spectroscopy extremely deep technique. It's a physics of weak forces in all definitions. And how different elements can be detected selectively without interference. So you have seen proton NMR, you have seen carbon NMR, you can record nitrogen, you can record chlorine, you can record deuterium, phosphorus, fluorine, and all other elements present in the molecule don't even interfere. So why in one NMR technique in which you have so much of control of such a selectivity, and how to solve sensitivity problem, because it is very, very sensitive. So from the time of it was detected till to date, many Nobel Prizes were given to people in two areas. One, who solved the sensitivity problem, and the other, who used this technique for a new application, only two broad categories. And what is the second dimension in NMR spectroscopy? That is what I'm going to focus in next 1 hour. I cannot promise that I can answer all of these questions, but these are enduring questions. It goes into the basics and fundamentals of NMR.
So if the online people are complaining, they cannot hear you very well.
Okay, can you hear me now? Okay.
The online ones.
Online one. Okay, my dear brothers and sisters online, please let me know whenever I am not audible. Okay, so this is what the questions are. And okay, so these are some of the books which Professor Tharuman have actually showed you. The third volume of this book, the third revision is in the process, so you will be able to see this wonderful book with which is focused on solving problems in NMR spectroscopy. And next, and I would leave this lecture for you, so you would be able to see some very good resources of fundamental questions which I asked you, and these are some of the responses in them. Next. And you know that NMR is everywhere. So NMR developed by physicists. Physicists were the first who developed NMR, were extremely unhappy on NMR, almost abundant NMR, never looked back, and then chemists adopted that. And chemists were very happy. And then biochemists jumped in, and they start liking NMR for various different purposes. And then medical community came and they said, okay, we're going to use NMR for medical diagnosis. So I will go back. So this is your NMR. This is 1.2 gigahertz. This is your MRI, magnetic resonance imaging, for animals. So a little mouse is sitting here, and we can have MRI of this little animal. We can have a whole individual going inside, and you can use it for a variety of different purposes. You know that MRI is animal. And why is called MRI? Because when this was developed, the name of the technique was NMR, medical diagnosis device. People didn't really like nuclear word. People were just so sick after this Hiroshima, Nagasaki, that they didn't want to listen this nuclear machine. People are placed in nuclear machine, though it has nothing to do with that nuclear. So the name was changed for political reasons, making it more acceptable of magnetic resonance imaging. So it has application in whole range of different topics. Next. And it is, after all, a spectroscopic technique, but very different one. I'll tell you how different it is by all definition. So it is studied like any spectroscopic technique, molecular structure and dynamics through the absorption, emission, scattering of light, electromagnetic radiation. This is what spectroscopic techniques are. Next. And this is this beautiful electromagnetic radiation case. Everything there is used in some form of a spectroscopic technique. Even very strong rays. So these are extremely high energy electromagnetic radiation. These are relatively lower, and then of course these are weakest radiofrequent electromagnetic radiation. Each one of them is used for a spectroscopic technique. Each one of them. So gamma rays, gamma spectroscopy, it's really used for detection of isotopes. X-ray is a very well-established technique. You look into the scattering of the electron scattering, the X-rays, UV spectroscopy, IR spectroscopy, microwave spectroscopy. How come on the face of the earth for literally decades and decades? Everything from gamma rays all the way through to microwave. Everything. You give electromagnetic radiations and you see absorption or emission of that radiation by your sample, by your method. Except radio frequency. Radio frequency for decades. UV and IR were detected at the end of the century of 19th century. X-ray was developed in early 1900, 1912, and Braggs developed the theory of it. Almost all the spectroscopic techniques were developed, though equipments were developed later, but it was already known that you take any of this electromagnetic radiation, you see an absorption or emission or scattering, except radio frequency. So next. This was the holy grail of spectroscopy. People could not, even very smart physicists were not able to understand what is spatial and radio frequency, why it is not absorbed, emitted, or scattered or whatever. Why radio frequency is not absorbed. You bring any sample and you bring radio frequency as electromagnetic radiation, you see no absorption on your chart, no absorption on your paper chart. Why it happens? A fundamental, basic myth and a problem in physics in which for many, many years, for decades, physicists were wondering why radio frequency is not absorbed. It is electromagnetic radiation. That IR, UV, X-ray, but why radio frequency is not absorbed? This was a holy grail of spectroscopy. And this was the basic architect of any spectrophotometer or spectrometer. No matter how complex they look, you can go to any spectroscopy laboratory, you will find lots of computers, you will find many screen touch, you will find many small buttons and everything, but essentially, this is what spectrophotometers are. 3 essential components. Electromagnetic radiation source, your sample holder, and your analyzer or the detector, whatever. This is what you need. So you bring infrared, you get infrared spectrum. You have UV, you get UV spectrum. You have microwave, you get microwave. You have X-ray, you get scattered, and you get this scattering diffraction pattern. You bring radio frequency, you bring electromagnetic radiation, you see some effect. You bring radio frequency oscillator, you have nothing there. Nothing at all. And that is where this Holy Grail started to work. Next. So you already know the advantages and disadvantages of very technique. Next. But NMR is a technique which was developed. Initially, the NMR phenomena was accidentally discovered in 1938. In a physics laboratory, 1 kg of lithium fluoride, 1 kg. I'll tell you what is the story of that. So name, nuclear magnetic resonance, is the most descriptive and most beautiful name of any technique. Nuclear, atomic nuclei. Magnetic, you need a magnet. You need to have an applied magnet. I will tell you why. And when you place your atomic nuclei, your sample, under the field of magnet or magnetic flux density, you see a resonance, and that resonance is detected by radio frequency. That is the description of the range. So if you understand the name of the technique, you understand half of the NMR. The theory of NMR is embedded in these 3 words: nuclear, atomic nuclei. Magnetic, because you cannot detect any radio frequency absorption without placing them under the applied magnetic field, magnetic flux density. And when you do that, you can see the resonance, which you can detect by using radio frequency. This is the name. Next. And this beautiful NMR spectrum. You like it? The first published NMR spectrum on 30 megahertz permanent magnet, 30 megahertz of ethanol was published. It was not the first NMR, because NMR of paraffin, lithium chloride, lithium bromide, water was detected before, but this was the first general published NMR spectrum in a journal. Isn't it beautiful? That made physicists absolutely upset. Physicists were so terribly upset. They said, what is this? We have been working on this problem since decades. We thought hydrogen will give us 1 peak, and we see 2 peaks. There is some chemical shift, you know. In a very negative sense, they said there is something wrong in the chemistry of this molecule. And that is the reason why 1 peak is split into 2 peaks. And at that point of time, physicists abandoned NMR. They said this is a bad technique. We don't like it because it gives 3 peaks instead of 1 peak for hydrogen. But then smart people like Salim Sabri, they said, good Lord, this is what we need, isn't it? We know always, we knew well before this spectrum that ethanol has 3 different types of hydrogens. So this is the only technique You just need UVIR. You just have these traditions of different states, nothing but absorption without any discrimination. Here you see the molecule, the three different types of protons showing three different peaks. They said, good Lord, this is the technique. Second, on top of it, they were also able to see that not only these were three peaks, but also proportionately they were representing the number of protons present under the peak, so-called integration. And they said, this is what we need. This is exactly what we need. And at that time, NMR was adopted by chemists. NMR become chemist baby. And physicists start working for chemists to develop better NMR, but not for them, because for them it was a hopeless thing. That is where NMR become absolutely wonderful technique for chemists. And it was in early 50s when NMR was adopted by chemists. Next. And today, if you record ethanol, it looked like that, essentially the same information. Three different types of protons based on chemical shift. You see, this word chemical shift came from physicists who said there's something wrong in chemical, which is shifting the peak. So this so-called insulting word by physicists are now used to define the microelectronic environment which make protons different from each other. So if you record NMR spectrum of ethanol today, you would see three protons, types of protons. You would also see the size or area under the peak, which represent number of protons, but also see the coupling. So you get three information, not one information, three information. What type of protons they are in terms of their microelectronics, how many of they are, and how they are coupled to each other. So second fundamental question comes to you now. Next. And since then, since the development in 1940s, when the first NMR were actually reported, till today, yesterday, Professor Taraman showed Earth field NMR spectroscopy, in which you don't need any magnet, you use Earth magnet, Earth magnetic field, tubeless NMR, LCMR, many development which took place right from the beginning of 1940s till today. Next. And this is endless, yesterday, Professor Taraman mentioned about fuel detector. Now, NMR machines are coming with three detectors. You can detect three different nuclei at the same time. So much of development took place in NMR. Next. But these are the people, who have started training physicist Isaac Rabi in 1938, not in 1944. He got the Nobel Prize in 1938. He was the first one who was, what he was trying to do was that with his group in the West Coast, in Boston, Harvard, he was trying to understand what happened to the element if you place them in magnetic field. And then you have different radio frequencies. With absolutely no intention, he used lithium bromide, placed it under magnetic field, brought different types of electromagnetic radiation, also radio frequency. We saw the absorption. Very first time in the history of physics, the radio frequency was absorbed by matter only because of the presence of magnetic field. And he was the first one who accidentally discovered that you can absorb radio frequency if you place your sample in magnetic field. Now, what is the magic? Okay, almost independently, person in Block, he was working in Stanford, he was working in Harvard, he worked on water, he worked on paraffin. After 4 years, they recorded very first time paraffin and water under the magnetic field can absorb radio frequency, and they then independently reported in 1944, independently in June 1944, that you can really absorb radio frequency if you place them under the magnetic field. Okay, so Nobel Prizes after Nobel Prize. And Professor Richard Dance was a very good friend of ours. He visited our center, died unfortunately years ago. He was the one who developed the concept of polarization transfer. He solved the mystery of sensitivity by using software technique, not the hardware technique. He was also the one who contributed in the development of concept of pulse spectroscopy. He got the Nobel Prize. Also, Professor Kurt Woodridge from Switzerland, he developed the application of NMR spectroscopy in biochemistry, in the field of protein chemistry, structural biology. He got the Nobel Prize. And then in 2003, these two individuals working in Nottingham used NMR spectroscopy, commercially developed a very high quality machine for application in magnetic resonance imaging. MRI, the technique was developed back in 1992, but they got the Nobel Prize in 2003. You work on it, you get the Nobel Prize. So NMR is much technique, which is perhaps you have most likely chances of getting the Nobel Prize. Okay. Thanks. So what happened was that accidental discovery, when you place your sample in magnetic field, then you see the absorption of radio. This is a workshop. Let me ask you why. Why only in magnetic field? You see the absorption. Why see absorption to begin with? Absorption of electromagnetic radiation takes place when you have two energy states, right? Because something will absorb energy and will go into high energy state. It means that only under the magnetic field, the protons or any nuclei, atomic nuclei, is capable of creating energy states, and without magnetic field, they cannot. So under the influence of a magnetic field, magic takes place. And that magic is that energy states are defined, so-called quantized energy states are defined, and those quantized energy states have energy difference. That energy difference matches with the energy of radio frequency. Magic is called Holy Grail and so on. That is where NMR becomes very, very different. First difference. For all other spectroscopy techniques, you don't need magnetic. In NMR, you do need magnetic. So the first difference between the NMR and other spectroscopy technique, the presence of applied magnetic field, ever-present magnetic field. Without ever-present magnetic field, you will not be able to see any absorption. Now, NMR spectroscopy, why you want to do that? Why you spend so much time on it? Because you have so many applications. You want to know the structure of the molecule. You want to study the dynamics, reaction kinetics, equilibrium. This morning, I believe Dr. Atia has already showed you some slides about the structural biology, circulation, plasma, different NMR spectroscopy. So, next. structures of big molecules of different kinds, drug designing, you can, and why NMR is different. In IR or UV are actually your action. You have a sample, you turn on your electromagnetic radiation generator, you see absorption, emission, stretching in 10 to seconds. That's it. And you get this spectrum. You get this spectrum. In NMR, it is so slow that you can literally play with the NMR signal for minutes, and in some cases for hours. So you have tremendous control on NMRs, on the spectrum and the results which you wish to obtain from NMR. No other technique. Electronic condition and UV from here to here. Absorption from vibrational frequencies, bond transition in IR. You see, and the difference between low energy state of UV and high energy state, from low energy state to high energy state, 144 kilocalorie per 144, about 130. In infrared, this vibration of bonds, stretching and different times. The energy difference is 4 kilocalorie. In NMR, is 0.004 calorie. Or, I mean, how insensitive, how weak that signal would be. It is not even close to 0.1 calorie. So extremely weak forces. The whole NMR spectroscopy is the physics of weak forces. So next please. And of course, you do a lot of these things. I don't want to repeat it. You get the structure, structure through chemical shifts, connectivity, stereochemistry, dynamics, a whole range of different things which we can do by NMR spectroscopy. We can run through that. You have seen the example of STD, maybe exchange also, but these are very, very efficient use of NMR. So fundamental of NMR, and that is, for the sake of repetition, I'm going to repeat many things. Inherently, this is a presentation which makes sure that I keep repeating many concepts. So it is completely embedded in our understanding. Next. So NMR is why not all atomic nuclei are NMR active, why they're NMR silent, and some of them are NMR active, because this is something which you don't really find in many books. Of course, you find this, but I'm going to tell you the reason of what you don't find. Odd mass or odd or even atomic number, even and even odd. So either the protons or neutrons, you know that very well. They have to be odd in number in order to see in a nucleus to be NMR active. You know why? Basic fundamental physics of that all atomic subatomic particles have to be paired. And if they are not in even number, they are not paired. And if they are not paired, there is an inherent dissymmetry in that nucleus. And if that inherent dissymmetry exists, that are the only nuclei which will have magnetic moment, spin, and nothing else. In all atomic nuclei which have even number of protons and even number of protons and neutrons, they have pairing of all subatomic particles, neutrons or protons. The nucleus is completely symmetrical in terms of phase charge balance. Anything which makes the charge balance symmetrical make this nucleus spin. So only those which have either odd protons or odd number of neutrons have this inherent dissymmetry which manifests as spin. And that is the reason why you will see only those atomic nuclei NMR active. Anything else which is not odd proton or neutrons, they have even number, they are NMR silent. No, so carbon 12, our beautiful carbon 12. You know, we love carbon 12 very much. This is our lifeline, our business, our salary. Carbon 12 is not NMR active. It's so sad, you know. It is so heartbreaking that carbon 12 is not NMR active. But carbon 13 is NMR. So good news is that almost all periodic tables, at least one isotope of every element is NMR active. So conservation, sympathy, God's help. And you should be very happy that you can theoretically study every element because at least one isotope of every element is NMR active. What about oxygen? NMR active or not? The normal oxygen is normal. No, but oxygen 17 NMR active. What about nitrogen? No? Yes. Nitrogen. Seven protons, 7 neutrons. Where it comes? NMR active or not? It is NMR active, theoretically. Right? And 14 is NMR active by this definition. Well, I'll tell you why it's not visible. There are some other reasons. Thank you very much. So go back to the previous slide. Okay, so this is the definition that in order for an atomic nucleus to be NMR active, it has to have odd number of proton or neutron, one of them. Here, both. And you see, when such situation exists, odd, then they have magnetic moment, and that makes them spin around their axis because of inherent dissymmetry which they have in their charge distribution. So this is called magnetic moment, and in the presence of magnetic field, they start precession also. So they have two movements. One is axial moment, the other is angular moment, which is called precession frequency or Larmor frequency. And now the question is, next slide. This is I think one of the most important slides in my presentation. The simplest one. This is the equation 2i plus 1. I is a spin quantum number, and this is the equation which I would really like to do. So hydrogen, which I call proton. Why I call proton? Because we're discussing the NMR active nuclei of hydrogen, NMR active nucleus, which has only one proton. So we call it proton. This is the reason why NMR, the NMR community never call it hydrogen, we call it proton. Okay, so what do you think? NMR active or not? Odd and even. Only one proton. Active? Okay. If you take this and use that number into this equation, how is going to be how many spin states? You just build that thing into this. How many? Two. So two energy states, two energy quantized states, alpha and beta. Deuterium. Deuterium, one proton, one nitrogen. Spin quantum number 1. How many energy states? 3. Carbon 12, 0. No energy state. Carbon 13, 2 energy states. Nitrogen 14, 3 energy states. I'm not really doing any magic. It's a very simple thing. This is the spin quantum number which physicists have calculated. I don't know how. I don't want to know about that. But they, this is a weak question. Sorry. Okay, so this is the spin quantum number. You fit that spin quantum number in this equation. You know exactly how many spin states. And oxygen 17. Brother Muhammad said that oxygen 17 is NMR active, right? Because why? It has 9 neutrons, right? 9 neutrons. Okay, so how many energy states? 6. And fluorine, NMR active, 2 energy states. You see, this is the reason why whole science of NMR is so sensitive. If there are 2 energy states, they call it dipole nuclei. Only 2 poles, right? If they have more than 2 energy states, they call quadruple nuclei. Quadruple nuclei. So deuterium is quadruple, and 14 is quadruple, F17 is quadruple, they are quadruple nuclei. Now I will take you to a very, very fine point in NMR spectroscopy. You see the difference between energies. The difference between the energy state is so tiny, very little, that this is the only spectroscopic technique in which both energy states are populated. No other spectroscopic technique has both energy state populated. UVI or Raman, you just name it, everything is on low energy state. You give radio frequency, everything goes up to the high energy state. Right? But in animal spectroscopy, the energy difference between two energy states is so little that both energy states are populated in physics of weakness forces. Think of it. If you have both energy state populating, and the difference between population is so tiny, which is your NMR signals, what will happen if you have more than two energy? If you have more than two energy states, the population difference is going to be further divided. And when population difference is going to be further divided, then you don't see the signal. Theoretically, even with the strongest magnet available, which is 1.25 gigahertz machine available, you cannot really see many of these quadruple nuclei because the population difference, which is certainly visible if you have two energy states, if you have three energy states, 5 energy states, 7 energy states, 9 energy states, the population difference is going to be divided so much that you will not be able to see the signal. That is the reason why N14, which is theoretically NMR active, is not visible. Because your signal is your population difference. And the population difference between not 2 but 3 energy states is so tiny that you will not be able to see any signal. So this is what happened. This is the magic, all atomic nuclei are magnets. Since they are magnets, they are vectors, they have direction, and if you have million nuclei in the absence of magnetic field, they are degenerated. They are oriented everywhere. Everywhere. You don't have energy state. Once you place them under the magnetic field, which was in 1940s, permanent magnet, in 1950s, electromagnet, and in 1970s, superconducting magnet. The development in magnetism was one of the reasons why animals become very sensitive. So applied magnetic field, you have an applied magnetic field, the magic takes place, and these degenerated, the randomly oriented nuclei orient themselves into either aligned with the magnetic field or non-aligned with the magnetic field or opposing the magnetic field. The aligned is called alpha low energy state. The ones which are opposing the magnetic field are the high energy state called beta. So that is where the magnetic field has a magic. That was missing, for decades, for 40, 50 years. This is what physicists were trying to understand why the radio frequency is not absorbed. Now, once we have a magnetic field, as the turbine was able to see that a magnetic field, when applied, you have two energy states. One is low energy state, the other is high energy state. Radio frequency is absorbed, the difference goes up, absorption is recorded, NMR appears on the paper of Oxford chart. That was a magic. So what happened is that under the influence of a magnetic field, as I said, initially permanent magnet, then electromagnet, and then superconducting magnet. The benchtop, the desktop NMR, which are now available from Booker and Varian, are actually electromagnetic. for the one, the strong machines are superconducting magnets. Next, please. So this is what actually happened. You have low energy state, you have high energy state, right? You give radio frequency, they move from low energy state to high energy state. The absorption takes place. The absorption is recorded. So NMR spectroscopy in 1940 was developed as absorption spectroscopy. And again, let me repeat next that next week. Next. That under the influence of magnetic field, two magics takes place. One, that NMR active nuclei divide themselves into 2 energy states, 3 energy states, 5 energy states, 7 energy states, just name it. They have population difference between 2 energy states. You bring radio frequency, the radio frequency is absorbed, the NMR signal is recorded, and that is where NMR spectroscopy was developed. So the second thing which happened is that when you place them in magnetic field, they also have a second movement which is called precision frequency. So not only they are moving around their own axis, but also as an angular, like Earth. Earth has its axis movement, which is responsible of day and night, and its movement around the sun. And that is so-called orbital movement, which is responsible for weather, air, and etc. The same thing happened. This is the movement, which is called precision frequency. This precision frequency is your NMR signal. This is exactly what your NMR signal is. You don't need to do anything else. You can directly measure this frequency. And this frequency is directly proportional to V naught. The stronger the magnetic field, the more this frequency would be, resonance frequency. And the proportionality constant is magnetic gyration. I will tell you about magnetic gyration. You see, magnetogyric ratio is susceptibility of various elements towards the magnetic field. Very simple term. When you have a permanent magnet and when you have coins of different types, you have coin of copper, you have coin made of iron, alloy, different types. Do you observe the same kind of attraction between the permanent magnet and all of them? Certainly not, because they are made of different elements, exactly the same philosophy. The different elements have different magnetic gyric ratio, and their susceptibility, their response, and reaction towards the magnetic field is 10 to the 8. That is where I will answer the question why you are able to detect one element in the presence of the other, and that is the only technique in spectroscopy in which you can select one element in the presence of the other without any interference of any other element, and you would be able to record that. Nothing else, but this is the only thing. In X-ray, we have a leading expert in the field of spectroscopy by X-ray. All elements come, UV, IR, everything. You see all elements which are present in the molecule. That is the only technique in which you can select an element without interference of the other element. So that is where, and that is because of magnetic gyration. And now, this is the second most important slide. Okay, your NMR spectrometer is a magnet, a big magnet. The one you have in the University of Jordan is 500 megahertz. It is about, how many Tesla? About 6? About 6 Tesla, or 5 point some Tesla. Tesla machine, 5 Tesla machine. If magnet is same, element can be different. You know, you can detect carbon-13, you can have fluorine, you can have proton, you can have nitrogen, you can have all of that. And hydrogen is 99% abundant, which is fine. The machine is about, these are different Tesla. So this is the magnetic ratio. I would invite you to think about this constant magnetic ratio for different elements. For proton is 267, for deuterium is 41, carbon-13 is 67, fluorine 19 is 251, different magnetic ratio. And you remember that slide which I showed you about the resonance frequency directly proportional to V naught. So at 1 Tesla, the frequency of this Laurent frequency of hydrogen is 42. At 1.4 Tesla machine, it is 60. At 2.37, which is about 300 megahertz, is about, this is 100 megahertz. And for 7.05 Tesla is about 300 megahertz. You can see. Carbon, you see a very different behavior because magnetic ratio is different. So 1 Tesla, the frequency is only 10.7 megahertz. For 2.35 Tesla is 25 megahertz. And 7 Tesla is about 75 megahertz. For fluorine 19, 1 Tesla machine is 40 megahertz. So the same, you use the same machine and different elements have different frequency because they have a different magnetic ratio. So when proton at this particular magnetic power, the magnet is resonating at 300 megahertz, the carbon-13 is resonating with 75 megahertz because it has 1/4 of magnetic radiation. Let me ask you a question. This is where NMR becomes absolutely selective. That is where you can select any of these and you detect only that. So if you select 75 megahertz radio frequency, you would only detect carbon-13. If you select 40 megahertz, you will only detect F19. If you select 300 megahertz, you will only select in the same machine. I'm talking about the same machine. You will only detect hydrogen. Traditionally, since except the first lithium compound, which was accidentally discovered to have NMR, all other experiments in the 40s and early 50s were done on hydrogen. And that is the reason NMR machines are, the nomenclature of NMR machine is based on hydrogen. So your NMR machine is called 300 because proton is resonating 300. It's not for all elements. If it is called 700, it is only because proton is resonating 700. If it is 850, it's because proton is resonating at 850 megahertz, not carbon, anything else. So traditionally a wrong nomenclature, bad nomenclature, but you can't change it because even people who are never detecting proton and they work on different other elements, they still use the same terminology of hydrogen because that was traditionally developed. So I really want you to appreciate the fact that how important it is that magnetic ratio of elements is making NMR the only technique in which you can detect one element without any interference of the second element. Next. So my question to you, what happened when nuclei or your sample is placed in the static magnetic field? What happened? Any response? It starts spinning. No. That's not Dr. Salim. I don't. Yeah. Any? Yes. All right. So, take that in the magnetic field, two energy states. So, aligned to the magnetic field or opposed to the magnetic field. Aligned to the magnetic field, low energy. Opposed to the magnetic field, high energy. What else happens? Only when you place your sample in magnetic field. What second thing happens? Yes. Second thing, they start precision, and that precision is called Raman frequency. Raman precision. Joseph Raman, name of the scientist, this phenomena of angular movement, precision frequency is further beyond. Because this tiny little magnet, which is movement around the axial movement, also second thing happens. Okay, so two energy states. One is moving aligned with the magnetic field. The other thing goes to the magnetic field. Two energy states. If there are 7 energy states, what will happen? I scratch my head. If there are 7 energy states or 9, okay, this is a critical question for confusing everyone. And or 6. 6 energy states. Not so logical, but not so easy to understand. But logically, 3 of them are somewhat alike, and 3 of them somewhat opposed to the magnetic. And they are in several energies. And if you have a T10, the dipole nuclei will finish. If you have 6 energy states, the population can 6 states. So tiny that you will not be able to see the signal. And this is the reason why, despite the fact that fundamental property of atomic nuclei to have odd mass or odd number doesn't really guarantee that you would have animal accuracy. It's only based on the energy state available. So this is where the second most important thing happened, and that is okay. Now I think this one is an important slide. Next. Next, please. No. Going back. Okay, what about other nuclei? You know, we've been discussing about protons. Proton is very dear to us. It is a beautiful nucleus. It is very close to the heart. 20% very more active. What about these? So we're trying to answer that. Nuclei with odd mass and odd or even number, they have energy state of half. It means they would have quadruple, dipole nuclei. They all are detected, right? So you would have no problem of detecting carbon 13, chlorine 19, P31, nitrogen 15, no problem at all. But when you have odd mass and odd number, like oxygen 17, you would have quadruple nuclei, more than one energy state. You know, detecting nuclear quadruple, even mass and odd number, 1915. The spin quantum number is 1. So you have 2 energies, 3 energy states, -1, 0, +1. It is NMR active, but not detected because of the competition. The natural abundance of N15 is very low, and N14 is not detected. NMR active with NMR atomic nuclei with even number are not detected at all. Okay, I have to go a bit fast. Okay, so what you will do if you wish to detect a nuclei with NMR, which is NMR silent, what do you do? A question. If you have an element NMR silent, that is because they will not give you NMR. You select a massive group. Is okay. Next. Okay, so this is what actually happened. That in the influence of a magnetic field, static magnetic field, we have, let's assume a proton spinning around the axis, and this is aligned to the magnetic field, and this is opposed to the magnetic field, 2 energy states. And next, the difference in energy states is directly proportional to 2 things: magnetic barrier ratio, which you cannot control. There's absolutely no control on gamma, also on B naught. So B naught is magnetic field. If you want to have more energy difference, why you want to have more energy difference? If you have more energy difference, the population difference will be higher. You know why? Because the movement of nuclear species between 2 energy states will be slow. There's going to be an energy barrier in between, and as a result of it, you would be able to see the more population. The more strong the magnetic field, the larger the energy difference, more the population difference, the stronger NMR signal. So the easiest thing which was done to begin with, when NMR was developed from permanent magnet, valent magnet, and superconducting magnet, you establish, you bring stronger magnet, you have more stronger signal, easiest thing, expensive solution. Because you have 500 megahertz. You have 500 megahertz, now you go to 800 megahertz, then you go to 1.2 gigahertz. And the prices are not only linear, they are exponential. So you get $1 million, 1.2 gigahertz is going to be like $5 million. So this is a solution from the sensitivity problem, because the stronger the magnetic field, the more energy difference, more population difference, more absorption of radio frequency, stronger the NMRC. So this is what the relationship between the V naught and the population is. Next, please. This is the critically important, very, very important, equation. Frequency is directly proportional to V naught to proportionality constant in magnetic direction. Everything is dictated by magnetic calibration. You cannot really have separate NMR for carbon 13, for nitrogen, or anything like this. You have the same NMR machine. Your magnet exactly identical. The only difference is that every element has a different magnetic calibration. As a result, it will have different absorption frequency, and as a result, you will be able to select that. Next. So, Raman frequency, right? And factors which affect Raman frequency. Right? Just like this. This is your Raman frequency. Now, the question is, we're talking about hydrogen. Only hydrogen. Think of all hydrogens. All hydrogens, same magnetic radiation, same magnetic field. Why they don't absorb at different at the same radio frequency? Why not? Why not? Physicists were so disappointed. Why there was no signal for all hydrogens? Why? What is that? Yeah, that is also true. That is also true. Everything is true. The fact is, if you remember that example, again, so we all are human beings, right? Homo sapiens, same magnetic calibration. Second, we are standing somewhere in North Pole, same cold weather, minus 7 degrees centigrade. Do we all see the same cold weather? Why not? Remember this very good stuff. I am wearing this very cheap coat. Somebody is not wearing any coat at all. And that is individual. Who same magnetic field, same magnetic calibration, but do we feel the same cold weather? Do all patterns feel the same magnetic field? No, because they have a different shielding. This is shielding. My electron code. This electrons around me is going to be different because of different reasons. Hybridization could be anything. And because of that, I may have a different type of a code. I mean, and the word is so beautiful, shielded and unshielded. Shielded against magnetic field and unshielded against magnetic field. So all have electronic shielding, and as a result of it, we will have different B effective. So B naught turn into B effective. B naught is same for everything, for every all elements and all members of the same element. But B effective is what we feel, what I can feel. And that is the reason why B effective is different for C of ethanol, CH2 of ethanol, and OH of ethanol. As a result, the frequency would be different. And when there would be different number of frequency, different absorption. Right? That is why different member of the same element show different resonance frequency. This is the reason why you see different signals of hydrogen in the same molecule depending on the sheet. Now, the difference between the radio frequency for the same element is only tiny, very little. It's not like they become carbon or nitrogen. Not that difference. The difference is only in part per million. Part per million. The difference is going to be 300. If it is a 300 megahertz NMR machine, it's going to be 300 0.1 or 299.056 megahertz or something like this. It's slightly different in the chemical shape, right? That is the reason why NMR is so powerful that it can detect the different members of the same element. Next, please. Next. Okay, then I told you the guess of energy difference. You know, to energy state alpha and beta, only 0.004 calorie. Very, very little difference. And it comes under radio frequency. So radio frequency is the only source which matches with this tiny energy difference. Next. And I already told you, NMR is also different because of this terrible phenomenon. You know, this is one of the most user part of NMR spectroscopy. Unlike, next please, brother. Unlike any other spectroscopic technique in which the electrons or energy state either is in low energy state or high energy state, NMR is both energy states are populated. And they have this terrible phenomena which is called induced emission. So the excitation and relaxation is taking place all the time. When you bring radio frequency, some of them absorb and go up, some of them transfer this energy and go down. So this induced elevation is going on between the two energy states, one of the most difficult phenomena in spectroscopic technique called induced elevation. But it actually takes place. NMR signal depend on population difference, and population difference is for Boltzmann distribution axis. Again, the name of the scientist, Boltzmann, who first discovered not NMR, but for other physics theory, the difference between two energy states in population difference is called Boltzmann distribution. Next. This is the question which you keep answering if you really want to understand NMR to the depth. In the absence of this, in the absence of magnetic field, in the presence of magnetic field, when radio frequency or appropriate energy drop, what actually happens? Next. Cortical nuclei, rapid realization, no NMR. Next. And I give you to the last part of my presentation. Next. I hope that by now we all appreciate that why dipolar and particle nuclei are different and why nitrogen 14 is NMR active, but it is not detected because it is a particle nuclei. And This is an equation which you always need to remember. Resonance frequency equivalent to gamma E naught applied magnetic field. This magnetic ratio is different for different elements. As a result of it, the resonance frequency is going to be different. Next. This is the most beautiful little diagram. So my nucleus is moving around the axis, but it's also moving angular momentum towards the direction or opposed to the magnetic field, called precessional orbital movement or Larmor frequency. If I want to detect this, I told you that this precessional frequency is dependent on electronic environment, isn't it? That is what you need. So if you place a detector here, you'll be able to detect the precession frequency and you're done. Fact is that you cannot really place magnet here because magnetic field is also in this direction. Everything is on this direction. Your detector is going to be burned in a few seconds. So in order to detect the signal, you have to bring them from z-axis to xy axis. And here we have this this xy plane where you have detector next. Here you can see going from up to bottom, next absorption is recording, next. This is what I told you about the effective. Next. Next. This is what we have discussed. Next. 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 You know, this is the schematic of NMR spectrometer all the time, always. The difference was only the magnet. So you have a magnet, which was in 1940s a permanent magnet, in 1950s electromagnet. Today it's superconducting magnet, radio frequency oscillator. This is your sample. Under the applied magnetic field, they are divided into two energy states. The proton, you have the Larmor frequency, you have absorb radio frequency. Your detector did not detect that, and you have this NMR spectrum. But the machine which you use for it, next, is like this. Terrible machine, you know. When I start using NMR, this is magnet. Brother Mahmood, do you remember? You don't, you know, you're much younger than me, so. Maybe Brother Salim probably remember that. Okay, so this is by magnet, electromagnet. So you see so much of these electrical cable, right? And here I have my sample inside that. Because without magnet, I will never be able to see any absorption. Okay. Then I have this radio frequency oscillator, like exactly like the old radio, where you have knobs. So I increase the radio frequency slowly. Why it is big size? Because everything was done standing. Yeah. So you have sample. You turn on the knobs like this. And as soon as you see the absorption is recorded on this paper chart. So this is a paper chart. This is a paper chart on which I see my absorption of it. So it's terrible. You know, it used to take several hours to record one NMR spectrum because slowly you need to move the radio frequency. Very slowly, because as soon as the radio frequency matches with the Larmor frequency of one of the protons, it absorbs, right? Now, very, very inefficient. Extremely painful machine. Next. Next. It's called classical or continuous wave NMR spectroscopy, absorption spectroscopy. Next. 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 Now, the NMR spectroscopy, the last part of it, it is no more an absorption spectroscopy. You don't need to stand like this. You don't need to turn on the off. You don't need to see absorption. You don't do that anymore. You know, you are absolutely free. You're happy people. Because you spend $1 million and you get a machine like this. This one. This was $2 million. Okay, so scary looking machine, right? First of all, let me tell you that these are two divers. Outside, liquid nitrogen, inside liquid helium, because magnet has to be cooled at a very low temperature of -279 Kelvin. And liquid helium is the coolest liquid available. So you have, once you get this machine commissioned, you give the electricity, you turn it off, and this machine is running forever until you keep the liquid helium. You spend about $10,000 for buying liquid helium, you're okay. The day when you don't have the money, and when you don't have liquid helium, the machine is trapped. Nothing, it's brittle, you know. So nothing very special. It is just a protection of magnet and operation of magnet. Inside liquid helium, outside liquid nitrogen, and right in liquid helium there is a magnet, and that magnet has a sample where you place the sample. Okay, next please. And that is the reason why this machine costs so much. This is absolutely the marvel of physics, you know, insulation sciences. You see, liquid helium -279 Kelvin. This is your magnet. Your sample is here. Just a centimeter away, the temperature is -278. Sample is at room temperature. What kind of insulation you need? Think of it, -278. Sample is at room temperature. The insulation which required is the real sun. That is the reason why, if you have the entire machine made of gold, it will still be cheap. The entire machine, if it is made of gold, pure solid gold, it would still be cheap because the money is not for the material, money is for the technology. Here, right a centimeter away from -278 Kelvin, you have your sample at room temperature, ambient room temperature, 25 degrees centigrade. And that is what this machine is all about. Next. Next. Okay, so this is the last few slides. Now, the entire theory of NMR spectroscopy. There is the only technique in spectroscopy where the theory has changed. UV was always used, you know, absorption of ultraviolet radiation. IR was always there. X-ray from the beginning never changed. You may have scintillation counter detector, you have electromagnetic, you have area detector, you have everything is same. The detectors, everything is changed, but theory is remain the same. Here the theory has changed. Now, the theory has changed because it is not an absorption spectroscopy. It's not even an emission spectroscopy. It's completely non-radiated signal. What happened is the theory has changed. Initially, continuous wave NMR spectroscopy, you have your signal, your nucleus, it absorbs, it goes down, you record an absorption. Now, what is there? That all of them, you know, the low energy state or nuclear species, these are vectors, they have a direction, and these are the vectors in opposed to the magnetic field. The resulting the difference between them is R0, resulting magnetic vector called bulk vectorization vector. What happened is that you don't use absorption spectroscopy, you excite them all by giving a pulse. That is what NMR is absolutely different. All theories, all books written in 1960s are completely useless. Completely useless because it doesn't explain the mathematics is useless, the algorithm is useless, everything is useless because what happened is that someone decided to use a pulse, exactly the pulse like we have. Radio frequency of a stronger bandwidth, excite them all together and record their realization. So instead of exciting them one after the other, you excite them all together by using a pulse. Next. Pulse means turning on the radio frequency for a time period and then switching it off. Radio frequency excite all nuclear spin at the same time. Pulse technique allows the recording of large number of NMR spectra. So what you need to do, your magnetization vector, your population difference, you excite them, you record their realization, you excite them, you record their realization, that is the NMR signal. Next. So you remember that I told you that radio frequencies, the absorption frequencies of different elements are different. Next. Next. Three things. Pulse. Pulse power, how strong the pulse is. Pulse width, how the pulse width is defined. Pulse power means which element you want to detect. You want to detect carbon, nitrogen, hydrogen, oxygen, water. Second, pulse width defines which portion of particular nucleus you wish to detect. The third, pulse duration. How would you like to have your intelligent vector term? Next. This man got a Nobel Prize for this concept. He made and see the sensitivity enhancement because you record more NMR spectrum, the better the resolution will be because always remember noise are random, signals are overlapping. Every time the noises will be random, the signal is going to be overlapping. As a result, you record more NMR spectrum, put them together, you get a better signal to noise ratio. And next. So you give pulses. Next. 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 And what happened is, if you give a 90 pulse, your magnetization vector or X-ray wave, radio frequency is absorbed, radio frequency is never emitted. It is a non-radiating process. The radio frequency, which is absorbed by the nuclear spin, convert into 5 different kinds of energy types. Temperature, vibration, electrical impedance, magnetic impedance, and one more which I don't know. Only detector, which is this detector, when this magnetization vector is passing through this, detects the electrical impedance. No radio frequency is detected. So it's not an emission spectroscopy. Absolutely not. It's not an absorption spectroscopy. It is a spectroscopy technique in which you detect the realization of different nuclei based on their inherent properties, based on electrical impedance And the signal is like this. The signal which you get from NMR spectrum is this beautiful signal. This one. This is the most beautiful signal you get because they are moving on the X-ray plane, they're going up, and all nuclear species give pre-induction decay. This is the time domain, and that converts into frequency by using Fourier transform. So this is the only technique. I leave you here at this point in time. I want to tell you that error spectroscopy is the only technique, concept, theory, and application has been over the last 60 years or 70 years. It is not an absorption spectroscopy. It depends, it has a tremendous control. We have this control of taking this magnetization vector, and by giving pulse, we can bring it to X-ray plane, we can bring it to -z, and then you can respond to the signal, which is non-dedicated, and it depends on the limitation of the NMR or the atomic nuclei the signal. And today, all NMR spectrum which you see are actually converted by Fourier transformation. Otherwise, they appear like this. Thank you. Thank you very much.
Maybe that they are now trying to absorb the information, to digest. It was. Yeah, all right, sorry.
Okay, this was my full course. The future of NMR is very interesting. The machine which is now developed doesn't really use radio frequency at all. The energy difference at 1.4 gigahertz is equivalent to near infrared. You don't need radio frequency. This would become an IR. Oh yeah, there's energy. You see, the energy difference is directly increase the magnetic field and the energy difference measured by until 1.35 gigahertz. It is the domain of radio frequency. It goes beyond. So this will be next 4 years in NMR is going to be an IR. Which doesn't require money in. So sensitivity was enhanced not by buying expensive, just changing their home. The doctor just deciding them together at the same time and then according to
Now, it will interfere with the microwave. It will interfere with the IR. This is a disadvantage.
No, you see, at the end of the day, what you're trying to do is trying to understand longitude. And that basic fundamental theory is as follows. When we have a stronger mandate, we would have more population, better system, and systems are recorded by any.
But now, like microwave, the rotation compounds will absorb, not due to like opposition of the spinning. It's due to like the rotation of the molecule, the microwave will absorb also the vibration, the bending. Now, the selectivity of the NMR is lost now.
Selectivity is going to be lost, but an example that we are not really recording the absorption of any radiation or any electron. They all are being record is selectivity. What it says that it is the IR is IR.
I'm sure that they have the choice. They want to hear more lectures. So I will ask you kindly to send us a link of more and recommend some more lectures for the students. I'm sure that they would like to hear more about NMR spectroscopy. So maybe you can share with me and I can, or we are on the COMSTECH side, yeah, to have some more video lectures of Professor Ata and Professor Iqbal and maybe many other scientists.
And a 40-hour lecture.
Yeah. Yeah. What I ever want to find more and more. Yeah.
What important is negative variation.
Just want to take this opportunity. Probably this is the first time
somebody who has written several books on the subject, somebody who understands very well. I've been teaching NMR many years. There are some questions which I was not able to explain to my students. Theories not explained them very well. Thank you very much. Prof, you know, I know nowadays you are very much difficult administration. It's very going to be increasingly difficult to find an answer. So, but the technology has improved. Recording lectures. I use the Camtasia software, where you're almost there. The students feel as if you are there because text, video, and it incorporates into the PowerPoint. Half of the slide will be there. You can learn everything there at this point can be good for stake ICCPS. Just make sure it's okay.
No, so every Saturday at 8:30, I deliver a lecture. Every Saturday.
So we need this Zoom meeting so that we can all attend.
Absolutely. It is internationally branded and whatever I can do.
So we'll be waiting for the Zoom meeting link and more videos to be shared with us and with the student. As you have mentioned, the same. I was really listening, and it was very interesting. Thank you very much, Iqbal, for this. Very informative for all of us, even the one we have been teaching NMR, but it was put in a way that we like also to attend it ourselves again. That will also help us to clarify the Selma, yeah, here.
So with the new electronics available, what actually differently, which is good enough for small molecules, rapid elimination. Um, the second question, with this high magnet, how difficult thinking about
When he leaves, that would be my facility.
the second question that so how is the molecule
project is about 75,000. So we do it at if they have themselves to low themselves and to get very good now for us hybrid animals, you can protein have number of things. Not only science, but also the nature, the clothing, the qualitative clothing. I would say that the hierarchy of animal word was
There are 12 of them.
We don't need to ideally I would say that we would have
Any more questions? All right. I'm glad to see that Professor is telling me that about 40 to 45 people are online attending with us this lecture. Most of them are students, young scientists. So we're so glad that organizing such a workshop will benefit this number. And we really thank all the online participants for their interest, for sharing with us and for being with us through this workshop. And we're looking forward also to see you with us tomorrow. Tomorrow we're having a very interesting program that will start with an online lecture from an eminent professor, followed by X-ray from Professor Kristen Fritzl, who is a well-known molecular structural biologist on X-ray. So, and the whole session actually will be given. We extended the time. We asked the Professor Firas to give him the full time, so that he will give also protein X-ray and some examples. And he will. He asked me that whether anyone who is expert in the field. I told him maybe most of us, we are not. So he's going to give a lecture, a teaching lecture that I'm sure that we are all going to benefit and we're all going to enjoy. So this is a change in the program that I wanted to make a note for that. And Professor Pira's lecture will be just after the lunch. So our program, we're having the panel discussion for 1 hour. This 1 hour is given to Professor Piras, and the panel discussion is open. Like now, we will see how long would you like to stay, and all of the trainers will be with you for any question to be then concluded by the closing ceremony, and to see how, just to listen to your remarks for improvement of the coming workshops. So we have a coffee break, but maybe while you're going to the hotel, you stop by if you like to have some coffee, tea, and just some cookies, because we'll have the dinner will be at Sheraton at 7 o'clock. All right, we will meet there.